for a start, here we have a picture of the Molonglo River, which runs through Ngunnawal country, which is where Jar and I are coming from. We're in Canberra at the moment. I wish we were all together in Melbourne. The Molonglo's in flood again today after all the rain we had last night, although I suspect not quite as much rain as Brisbane, where Felicity is. So anyway, this is a reminder to acknowledge the traditional owners of our country and pay our respects to the Ngunnawal, Ngambri and Ngarigo people. And the sun's shining outside now, so hopefully things will dry up a little bit. So my name's Lindell. I'm an evolutionary biologist, and I do feel a bit silly being an evolutionary biologist starting off a linguistics masterclass. I do feel like a fraud because I'm not a linguist, but the point of this particular session is to talk about the way that an interdisciplinary projects have evolved. So it's the intersection between evolutionary biology and linguistics we're talking about, and the way that we've worked together on some language evolution projects. So I'm an evolutionary biologist, so I'm pretty sure that everyone attending this session is going to know more about linguistics than I do, but I'm here because I've been working on how we can use ideas and analysis tools from evolutionary biology to answer questions in linguistics. This is something that Sarah and I have been working with Felicity on for about 5 years now, and with various projects. So I'll mention some of those projects today, but most of today's session will focus on Felicity's work on grungy creole, which you'll hear more about later. So let me see if I can get this to work. Oh, good. So as I said, I'll be starting with just a very general introduction and sort of giving a bit of an overview of some of the ways that analytical methods from evolutionary biology can help answer some questions in linguistics. I'll just point out now that probably the most obvious place where there's been intersection between evolutionary biology and linguistics is in phylogenetics, and that's the one thing we're not going to talk about today. Why? Well, Simon Greenhill and I did give a language phylogenetics summer school masterclass a couple of years ago, and anyway, the work that we've worked on together, the three of us, is more around the modelling of change in populations. Then Felicity's going to come in with a more detailed look at these projects, in particular describe how you would collect data that works well in these sort of evolutionary methods. And then Zhao will come along with talking about how she has adapted the methods we use in evolutionary biology to work on linguistic data. Now, all of these are just going to be talking rather than hands-on, but please do interrupt as we go, particularly for interdisciplinary stuff. It's important that if there's ideas as we go that aren't well explained or that you don't get, it's important that you interrupt us before we go on. So please do feel free to pipe up. So you can type in the chat. I might not be able to cover the chat while I'm talking, but Felicity and Jar might be able to, or just turn on your mic and say hi. So actually, there is a very long history of evolutionary biologists and linguists working together. It goes way back to the start of evolutionary biology. So I don't know if anyone recognises this diagram is the only diagram from Darwin's Origin of Species, which is a hypothetical phylogeny displaying his sort of proposal for how it is that lineages would, the variation within lineages would build up and lead to diversification and eventually lead to many species from a single ancestor. Now, at the same time that Darwin was working, Alger Schleicher had been building language trees. So this is his groundbreaking Indo-European phylogeny. Now, he'd been doing this anyway, but when he read Darwin's work, he was absolutely inspired by it, and he saw the connections between the two fields. And in particular, and also the evolutionary biologists clearly saw the connections between language change and biological change, and they would often use language evolution examples to make the Darwinian hypothesis more plausible, because they could language change was something people could see happening in real time. They could see variation arising in populations and language changing over time, unlike biological evolution, which is much slower. And Schleicher too said that not only are the mechanisms of language change similar to the mechanisms of biological change as Darwin had described them, but in many ways language evolution is a much more tractable case, and Schleicher actually felt that he could provide a better test and better proof of Darwinian evolution using language data than biological data. So there has always been a link between the two fields, and at their heart, language change and biological evolution, our understanding of them are built on the same fundamental principles, and very often we find that we can use similar analytical tools, although not always directly. Often they need adapting, because I don't want you thinking we're claiming that biological evolution and linguistic evolution are the same thing. There's some very fundamental differences, and so you often need to adapt the methods to work with language data. So we feel that, this has always been an area of rich interdisciplinary exchange, and that some of the methods developed in evolutionary biology can be very useful in language evolution. So the reason I started working on language evolution as a biologist is that very often we're asking similar sorts of questions. So the questions I was asking about the rates of molecular change, Simon Greenhill, who I first worked with, was asking the same sort of questions of the rates of language change, and we'll see some of that later. So this is where the shared perspective starts. And biologists have had a long time to develop tools that answer the challenges of studying evolutionary systems, because there are some very specific challenges that don't apply to other forms of data. Okay, so some of the questions I'm going to give you an overview of in this first hour of the class is how do we characterise individual level variation and how it contributes to population level change? How is that variation shaped by the environment of speakers? What kinds of speaker populations change faster? Do some languages change faster than others? And then the broad scale picture, which is what stimulates the diversification of languages, and are there correlates of language loss? So we're going to be applying tools from biology to characterise individual level variation, to characterise the way that populations change over generations and over longer periods of time. And then we're going to show how these evolutionary methods developed in biodiversity can be used to understand language diversity. Now, those first two areas will then be followed up in more detail by Felicity and Jar. The last part really won't flow on to the next two talks, but I still think it's worth covering because it is an area where evolutionary biology ways of thinking really are useful in understanding language diversity. So that will just be a brief overview of some ways of looking at that. So let's start with how we characterise individual levels of variation. So most of the examples we're going to talk about today revolve around Grungy and Grungy Creole. These are Australian languages. I'm not quite sure who's in the audience, so I don't know what background knowledge people have. Felicity is going to talk more about this later, but I'll just introduce it by saying that this is an example of rapid language change, where the traditional language of the area, Grungy, is now merging with a newer Aboriginal language called Creole. Now, Grungy mob are famous in Australia for running one of the most important strikes in the history of Australia, when Grungy mob walked off the cattle station on which they were essentially slave workers, unpaid involuntary workers, and demanded to be paid. And this then morphed into a land rights claim. And so you can see the Prime Minister at the time giving a symbolic handful of sand to Vincent Linguiari to symbolise this land rights claim. And so Felicity will tell you more about Grungy Creole, but just to give you a bit of a taste of that, here is a short video with Cassandra Algie speaking in Grungy Creole about some of this work, and I'm hoping you'll be able to hear the sound.
Madarat ngomber belajar, cara kat ngalun jauh na. Kurang jam cil jangan ada banyak, paling changing jamak pani. Kalau in cara kat, tu pun macam mana jajuan jauh ji. Kalau gue ada cara kat, saya maximum. Kurang je creole dan English. Nyangai kan look kan changing jang kama jalan. Keep ngerban langut jang kama punya ni, and song block. Malu work jang kama. Jauh aja, langut kaji, nambah kaji, and biology. Only always still cara kapnyang mga ada pancaro nyala.
Okay, so this is a case of rapid language shift because this community started with the older generation speak speak many languages, but there are two languages fluently, Grungy and Creole, and they could swap between the two. But the next generation, their children, the adult generation, they now mix up Grungy and Creole, but different people will use different mixes of the elements of both languages. But their grandchildren, the younger generation, now speak a mixed language that has incorporated some elements of Grungy and some elements of Creole in a more codified mix. So for every member of this speaker population, there are effectively at least two variants for any linguistic feature. So if you want to describe this bird, you might call it a mukmuk, or you might call it an owl. And again, if you want to say you were Sikwan, or you might say Junga, or you might use a different form of the past tense, depending on whether you use the Creole or the Grungy version or both. So you can imagine for each person in the community, they might use both versions. They might use the Creole of one and the Grungy of another. And so we can characterize which variants each individual speaks. Now, most sociolinguistic studies might use one or two or three elements like this that vary in the population. The amazing thing about Felicity and Cassandra's data is they have collected this data for 174 variables, including lexicon, grammar. And when Jar and I saw this data, we got extremely excited as biologists because this looks like the kind of data that a biologist is familiar with. So we look at this and it looks like a genotype. So a genotype is when you do a sample of genetic variants in the genome at genetic loci, particular genetic code where you know there's variation in the population. And you record for an individual for each of those sampled variable loci, which particular genetic variants they have. So we somewhat boldly call this the lingua type, the idea that you have a whole bunch of linguistic variables, language elements that are known to vary in the population. And for each person, you record which one of those variants they use. And for most of them, there are two variants, but for some, there are more than that, because there might be multiple Grungy or multiple Creole variants, or there might be innovative elements. So not only do they have a very large number of variables, but they also have a very large percentage of the population. So there are at least 78 speakers that have been coded for all of these variables. And we know some things about those speakers so that we can describe their age, which generation they belong to, which family they belong to, where they live in these communities, whether they grew up with a fluent Grungy speaker in their household, and how much schooling they've done. So the lingua type describes where in a multi-dimensional language space an individual speaker sits. So you can imagine for each language variable describes an axis of variation in their own language repertoire. And then these environmental factors can move people around in that space, and we can ask how things like schooling or growing up with a fluent Grungy speaker changes your occupation of that language space. So Felicity is going to describe in much more detail how you collect this kind of data. But let's just have a look at what you can do when you have it. So this is Jar's beautiful multidimensional language plot, where each one of these shapes is a different language variant. And we can see, for example, how different groups within the community distribute in this language space. So the older people tend to use a different characteristic set of language variants than their children or their grandchildren. We can also look at where particular variants fall in that space. So only the younger members of the community will call this a cane toad, rather than a ngakpan. And can I just say, you will notice I can't speak Creole or Grungy, so my pronunciation is going to be horrid.
Actually pretty good.
And only the older people will call this a mali-mali rather than a butterfly. And so we can look at the way social factors move people in this space. We can look at the way different language variants occur in this space. And we can ask what environmental influences move people around in this space. Now, Felicity will talk about this in more detail later, but just to give you an idea of where we can go with this, we can ask what the effect of environmental aspects are on language. For example, individuals with more years of formal schooling, which in this community is always in English, will have less Gurindji variance in their own personal linguotype mix. And yet people who have lived with a fluent Gurindji speaker, a grandparent, will have more Gurindji elements in their personal language mix. We can also use this approach to look at the way different classes of language variants are shaped by the environment. So, for example, we can show that perception variants, that is whether you understand a particular language variant, are retained in the mixed language longer than production variants, being able to actually use a particular language element for Gurindji. And we can also show that this effective formal education on reducing the amount of Gurindji in people's lexicon has a stronger effect on grammar than it does on lexicon. And perhaps they're not surprising results, but it shows the power of this approach to look at these patterns. Okay, so we'll see in more detail later how we characterize this individual level variation. Now we can use that to ask what influences the way those variants change in frequency over generations and therefore drives population level divergence. And there's two things I'm going to introduce here. One is Wright-Fisher models, which Shah will talk about in much more detail later. And then I'm also going to look at sister pairs analysis, which really is more part of diversity, which we'll just mention so that you know that it's there. Okay, so given that this mixed language is formed from two sources and there are two variants for every language element, we can ask, is the mix that you end up with in the third generation, just a random sample, you might end up with the Gurindji variant, you might end up with the Creole variant, or is there a bias towards some sort of class of variants? Now, this question about whether from an initial pool of variations, some are more likely to end up in subsequent generations, this is a question that evolutionary biologists are very familiar with. And one of the major tools we would use to answer that kind of question is a Wright-Fisher model. And here's a lovely picture of Sewell Wright with a guinea pig, which were his first love. He did his population genetics on guinea pig coat colours, and this is Ron Fisher. So if you talk to an evolutionary biologist, they will describe a Wright-Fisher model in terms of population genetics, that it makes assumptions about heredity and generations. But really what this is it's a generalized model for any situation in which you have a serial sampling procedure. So a serial sampling procedure is that the variants available in one generation, you get some but not all of those and start the next generation with them. And you get some but not all of those to start the next generation. Why is that interesting? Well, any serial sampling procedure can actually lead to change even if there's no bias towards one kind of form or another. So imagine a serial sampling procedure where we've just got a jar of different coloured marbles and we put our hand in and pull out a handful of marbles. And then we throw that into the next jar, and then we make up the volume of the jar in the same proportion. So in this case, we just happen to have got a few more blues than reds. And so our jar has now got, just by chance, more blues than reds in it. Now we can repeat that procedure, and maybe next time we'll get more reds and blues, or maybe we'll get more blues than reds again. Now, in a serial sampling procedure where each sample samples from the previous generation, it's possible to get a run of changes just by chance. You just happen to sample more blues than reds and you end up with change, not because blues were better, but just by chance. Why is this important? Because if we want to interpret patterns of change over time, if we want to say, there was actually a bias towards this kind of change, we first need to work out what kind of change you could get just by chance, not because either variant had a greater chance of making it to the next generation than another, but just because no sampling procedure is perfect. Now, Jia is going to talk about how she has adapted the Wright-Fisher model to work with language data, but let me just quickly give you an overview of how it was applied in the case of grungy Creole. So for any element of the language, there might be a grungy variant, there might be a Creole variant, and there might also be innovative variants that are not found in either source languages, but are new innovations in the mixed language. So for each of these language elements, we can say how many people use either one form or both forms, and we can characterize the frequency in which they're carried in the population by speakers. So here in the first generation, we can see that there's, sort of fairly even use of the Creole and grungy forms, but by the second generation, for the same item, there's more Creole and less grungy, and by the third generation, the child generation, the grungy form has disappeared, and only the Creole form for motorcar is used. Now, of course, that's just one element. Here we've got a whole bunch of lexical elements. There are also grammatical elements. Now, when a biologist looks at this, the words we would use to describe this, and I know these have different meanings in linguistics, we would say in the first generation, the population is polymorphic. There are many forms of the same language element. There are multiple variants. So this is not referring to the morphology of the terms themselves. We would then say that there has been an increase in the frequency of the Creole variant at the expense of the grungy variant. And then when the grungy variant is lost and there's only the Creole version left, we would say that the Creole variant has been fixed. We've lost the grungy and innovative variant. So now there's no more variation. So the next generation, if they sample from the child generation, all they can sample is the Creole variant. The grungy one has disappeared. So now we can look across all the different ones. And if you just look at one, you don't know whether that is just due to chance or whether it's because the Creole version was preferred. But if you look across all of the elements and you say, what's the general pattern? And here's the grammatical items here. Now, even just by looking at this, you can see the amount of blue is increasing. And in fact, if you model this, you can show that the Creole is increasing greater than you'd expect by chance. So there really is some kind of preference for the Creole variants. So then you can say, why? Why is it that Creole variants seem to have an advantage over grungy variants in making it into that mixed language? Now, Felicity is going to describe this in more detail, but this is just to illustrate what you can use a Wright-Fisher for. Felicity has coded all these variants by morphological complexity so that we can ask, well, one thing people often will say with a mixed language, with a language contact situation is that you're more likely to end up with the simpler variants. And this kind of makes sense. If you're in a population where you have two variants of any given thing you want to say, wouldn't you consciously or unconsciously tend to use the simpler form? So Felicity coded these for complexity, which she can tell you about later. But then we can show, using the Wright-Fisher, that actually there's no significant bias towards adopting simpler elements, that it doesn't make you more likely to get into the next generation if you're a simpler variant than if you're a more complex one. And you can actually see by looking at this that sometimes a complex Creole variant will be fixed over a simpler grungy variant, or sometimes a complex grungy variant will be fixed even when there's a simpler Creole variant. So it's a way of testing. Now, obviously no way of defining complexity is going to make everybody happy, and we can have that conversation, but it's just to show the kinds of things you can do with these tools. It's very hard to show selection for language elements. So this is one way of doing it. Okay, so now looking at that population level, at this point I'm going to briefly look at some of the diversity-based or macroevolutionary-based approaches we take. Now, these are not going to be covered later in Felicity and Jar's talk, but the reason I wanted to talk about them is this is somewhere where techniques developed in evolutionary biology really are quite handy in looking at historical linguistics and comparing rates and patterns of change across languages. So I just want to give you a quick overview of some of the ways these can be used, and very happy to answer questions afterwards, just so that you can see that there are these handy ways of doing things out there that overcome some of the problems that linguists have known about for a long time. Okay, so here's a question. We've looked at the way change occurs in populations, but what kinds of populations do we expect to evolve faster? So there's been lots of different theories that people have talked about. So on the one hand, people have suggested, well, obviously a large speaker population, you're going to have more speakers who are innovating. There's going to be more new language variants being put into the language every generation. So maybe they will change faster. On the other hand, you might say a small population, any given innovation can spread throughout the whole population much quicker. So maybe they should change faster. Now, it has been said that large populations might have more reliable language transmission. You've got more expert teachers to learn from. And that maybe small populations are much more prone to stochastic loss of language elements, because, you know, it only takes the loss of, for example, one expert elderly speaker to lose some language elements, which of course has been one of the really terrifying things about COVID, is the idea of losing those expert speakers from populations. But it has also been suggested that large speaker populations might actually lead to simplified language, because there's a greater diversity of speakers. There's more L2, second language speakers. The population might be in contact with more different other languages, so there might be a loss of complex language elements. Whereas it's been suggested that smaller populations, and particularly isolated smaller populations, might actually be fairly conservative against language loss. They might have more reliable language transmission. So you can see there are all these different predictions. So how can we actually test this in the real world? Now, what we can't do is assume that the same patterns we would expect from genetic evolution will apply to language evolution, because this is one of the cases where we can't assume the same processes. Why not? Well, if we take a small group of people and we take them off and we start a new society on another island somewhere, and their language is going to evolve, we know that that small group of colonists cannot carry all of the genetic variation of the ancestral population from which they've come. Why not? Because each person can only carry 2 genetic variants. And so that boatload of people we're starting a new society with, most of the rare genetic variants in the parent population aren't going to be included in that small sample. And if they are, then they're suddenly at much higher frequency. They're not rare anymore. In the next generation, they're going to be at much higher frequency. So genetic, if you start a genetic population from a small handful of individuals, you can't bring all the genetic variation with you. So there will be a founder effect, a rapid loss of variation. But it is at least possible for that boatload of people to carry all their language variants from the parent population. Not that they necessarily will do, but there's no physical impossibility like there is for genetics. It's possible you could have a boatload of expert speakers who have pretty much all of the language variants. So we can't assume that language will undergo a founder effect in the same way that genetics must do. So we need to test it. How are we going to test it? Well, you know, I mean, the nice way to do it would be to set up a replicated experiment. Get a whole bunch of people who all speak the same language, split them into different populations, large populations, small populations, go and stick them in isolation somewhere and come back in about 1,000 years and see which ones have changed the fastest. So, you know, it's fairly hard to get funding for a millennium-long experiment, but luckily, let alone getting it past an ethics committee, but luckily we don't actually have to, because this experiment has already been run for us. We already have sent boatloads of people off to different islands to start new language groups. And we can now go back and say, well, do the larger or the smaller ones have the faster rate of change? But, so for example, there was once a bunch of people who, for convenience sake, will say spoke a language called Marquesic, and they went and they started a population on the Marquesas and a population on Mangareva, which is a much smaller island, and the island size determines the carrying capacity of the population, so how many speakers you have of each language. So we can compare these two languages and ask which one has undergone a greater rate of change. Now, obviously for one pair of languages, that doesn't really tell you very much. There could be just something weird about Mangareva that happens to increase their rate of language change. But if we get a lot of these pairs, and if each one of them represents a different trial, basically an independent experiment where you get a bunch of people and split them into two populations, then if we get enough of those, we can start to ask if there is a general pattern. Is the smaller one usually the one that has a faster rate of change? So we compared pairs of Polynesian languages that are thought to be each other's closest relatives. So now we have the languages, we need a way of comparing rates of change. So to do this, we sort of repurposed a somewhat archaic test from rates of molecular evolution that was devised to compare rates of change in DNA sequences. And we used a database of basic vocabulary. So this is like a Swadesh list where you have words that are found in all different languages, like hand, foot, grandmother, water. And you ask whether the two languages have cognate terms that are shared with the ancestral population. So for each sister pair of languages, you might, so, and again, I'm going to say I don't speak either of these languages, so for anybody here who does, I apologize for my pronunciation. So it's quite common for Austronesian languages to have something like lima or rima or ima for hand and for five. So where we see that, we can be fairly sure that's the retention of an ancestral form in both of those languages. But if we have something where one of the language pair has the ancestral form, but another one has a very different form, we can assume that that cognate was present in the common ancestor, but then lost in one of that pair, which then gained a unique form. And there might also be cases where there is a word from the language family, like many languages have u'u or susu for breast or for suck, but Marquesan in addition has another word which is not cognate, so we can say they've retained one word and gained a new one. So this way we can separate out patterns of gain of loss into related languages by comparing them to the forms in the language family. So when we do this and we model the expected rates of change along these sister pairs, we find that larger populations gain new words at a greater rate than their smaller sisters, and smaller populations lose words from basic vocabulary at a greater rate than their larger sisters. And we were very excited by this result because this is actually what you would expect from population genetics, that small populations lose variants, but large populations are better at gaining new ones. So we were very excited about this, but of course this is just 10 pairs of Polynesian languages. So we then set out to test it for a wider range of languages, for pairs of languages across Austronesia, for Indo-European languages, and pairs of languages from the Great Bantu expansion. And what did we find? We found that for Indo-European languages, again, we see this pattern that smaller speaker populations have a greater rate of loss. And yet there was no other significant patterns with population size for Austronesian or Bantu. So I think this is a very important point to make because it tells us a couple of things. One is that there are significant and consistent patterns with population size in languages, or at least there can be, but they're either not universal or they're weak enough that they're overridden by other factors or that we can't always detect them. So there's certainly that potential there, but hopefully this has illustrated how comparing pairs of languages, if you can get enough pairs, can reveal some general patterns. Okay. So now I want to look at the way that we've adapted methods from macroevolution and macroecology, broad scale patterns of diversity, to understand the way language diversity is generated and some of the processes currently threatening language diversity. And really the point, all I want to get across in this part is just these are some of the sort of mental toolkit we use in answering these questions. So take one example. Why is it that when we look at a map of language diversity over the globe, for a biologist, this looks very familiar because it looks like a map of biodiversity. The same areas of high language diversity are also areas of high biodiversity. So this is a map of bird species diversity. So why is that? Is biodiversity causing language diversity? Is language diversity driving biodiversity? Or is there some other link between the two? Now, as an example of the challenges of asking these kinds of questions, here is one particular hypothesis that says biodiversity drives language diversity, which is that the reason there's more languages in the tropics is because there's more parasites in the tropics. What on earth have parasites got to do with language diversity? Well, here is the hypothesis that has been published by Randy Thornhill, Corey Fincher, and a bunch of other people. The hypothesis goes like this. And they actually implicate a lot of cultural variation as driven by parasite load. If you're living somewhere with lots of parasites, then your population will do better if you have cultural features that reduce your contact with your neighbours so that you don't pick up their diseases as well. So in other words, there'll be cultural selection effectively for xenophobia. This reduced contact between populations will lead to a cultural isolation. That means that your language will develop will more rapidly diverge from your neighbours because you're not sharing the language, and this will lead to greater language diversity. And as you can see, when you plot the number of languages per country against the number of parasites, you get this beautiful linear relationship. But the problem is that parasites, like most biodiversity, have a latitudinal gradient. There's more in the tropics. Languages also have a latitudinal gradient, and anything that varies with latitude will correlate with anything else that varies with latitude, even if there is absolutely no causal connection between them. So it's actually extremely easy to get a significant correlation between cultural traits, language diversity, and environment, because all of these things have a latitudinal gradient, so they'll always produce lovely linear graphs. So the main message I want to go across here is if you ever see a graph that has countries as data points and cultural evolution or linguistic diversity or any other aspect of language and environment, alarm bells should ring, because it's very easy to generate these relationships. So to illustrate why, let's look at a slightly quirky example, although the connection to language diversity will be obvious in a minute. Why is it that when you think of hot, spicy food, you tend to think of hot, steaming countries, and colder countries tend to be associated with much more bland food and fewer spices? Well, in the 90s, people published a hypothesis that said that if you live somewhere with a high temperature, you're at higher risk of foodborne infection. So this is higher parasites again. And therefore, any culture that tends to add antimicrobial ingredients to their cooking will lose fewer people to food poisoning, and therefore will do rather better. And therefore, there's selection for spicier food where there's a higher risk of food poisoning. So perhaps ill-advisedly, we set out to test this hypothesis and collected a really stunning amount of recipe data, which was a lot of fun, but took a lot of time. And you can indeed see that if you plot spice use versus temperature, you get this lovely linear relationship. But again, alarm bells should ring whenever you see countries or cultures or areas plotted in this way. And actually, if you take into the account the fact that nearby countries will have similar climates, and they'll also tend to have similar cuisines, right? If you travel from one country to another, you'll get less variation in cuisine than if you hop from one side of the globe to the other. That's all you need to generate these relationships. And if you take that into account, there is no significant correlation, nor is there a significant correlation between spice use and parasites. However, even accounting for the similarity between neighbours and relatives, there is a significant relationship between the average number of spices per recipe and the number of deaths on the road. Why is this important? Because it's already been shown that linguistic diversity correlates with traffic fatalities. So you can see where I'm going here with this. Language diversity is correlated to spicy food. And therefore, we can develop a new hypothesis of language diversity, which is if you live in a hot country, you eat spicier food, which causes you to have traffic accidents, which somehow causes you to have more. I'm not quite sure on the last step there, but obviously still needs a bit of work. Anyway, the point is that it's very, very easy to generate these correlations, but you must be very cautious in interpreting them. Now, here's a far more sensible hypothesis for linguistic diversity. Published by Daniel Nettle, and he said that if you live somewhere with a longer growing season, so for more of the year you can grow food or gather food, you don't have to store food over winter, then small groups can be reasonably self-sufficient, they can occupy a smaller area and they won't depend on their neighbours for support, and so they can develop in their own way and their own language. And this would lead to more languages per unit area. Now, this is a very sensible hypothesis, but again, you can see alarm bells should ring when there's countries plotted on the graph, because you can see there's some very clear clustering. So maybe, you know, these guys up here, sure, they've got longer growing season and more languages, but there's so many ways in which they differ from the guys down here that have short growing seasons and fewer languages. So we have to take this into account in testing this hypothesis. We can't treat countries as independent data points that can be used to test a hypothesis, because neighbouring countries and related cultures will be similar in so many different ways that it's hard to tease apart causal relationships from spurious relationships. Here's an example of why this matters. So there was a paper published a few years back saying that although mammal diversity and language diversity show similar patterns in New Guinea, they show opposite patterns when you consider threatened languages and threatened species. Because threatened mammal species in endangered mammal species tend to be concentrated in the highlands, whereas threatened languages, there's much more threatened languages in the lowlands. Now, the problem is if you take every square of New Guinea as if it's an independent test of this hypothesis, then every time you sample a lowland square, you'll get high language endangerment, low species endangerment. And every time you sample a highland square, you'll get high species endangerment, low language endangerment. It's only a single comparison. It's one thing that's making the highlands that way and the lowlands as they are, just the cultural and historical differences. But when we keep sampling them, we generate a false, we generate more data points than we actually have. So treating grid cells as independent is pseudo-replication. It's basically photocopying the same data point over and over again and pretending you have lots of data. When you take this into account, there's no relationship between the number of threatened mammal species and the number of threatened languages, because it's all driven by that one single pattern. Okay, so if we want to study patterns of language diversity and patterns of language endangerment, we have to take into account the fact that many variables correlate together. For example, spicy food correlates with traffic accidents, even if there's no connection between them. We have to take into account that nearby cultures and nearby countries will share many things in common, particularly in their environment and their history. And we have to take into account the fact that related languages, related countries will have many aspects in common, even if those aren't correlated together. So in order to study what drives language diversity, we collected as many relevant variables as we could. So it's often been suggested that hilly country with a lot of altitudinal variation will tend to stimulate language diversity because it will isolate people and stop them from being in contact with their neighbours, same as landscape roughness. And it's also been suggested that rivers are a universal determinant of language diversity. We also put in aspects of climate that are known to correlate with both biodiversity and have been suggested to correlate with language diversity. We look at aspects of population, so not only the speaker population size, but also the density of people in that area. And then aspects of biodiversity using representative species to represent how species rich an area is and what kind of biome it is. Is it forest? Is it grassland? And so on. And when we do this and we have Jia developed an analysis that takes into account the proximity between samples as well as the relationship between languages, we find that Daniel Nettles' hypothesis is indeed well supported, because language diversity is significantly related to the length of the growing season. And the growing season is related to the minimum speaker population size in an area. So instead of all of the languages being smaller where there's high growing season, it means that the smaller groups can persist in areas with long growing season. Surprisingly, we didn't find any signal in the data for isolation of language groups increasing diversity. So altitudinal range or landscape roughness doesn't seem to correlate with language diversity. But as has been suggested previously, river density correlates with language diversity, but not by chopping speaker groups up into smaller groups, but again by supporting a smaller minimum speaker population size. So again, this suggests that rivers are a resource that allows smaller groups to persist. So this allows us to not only identify correlates, but also to rule out some kinds of directions of relationships. So it's not biodiversity causing language diversity, it's not language diversity influencing biodiversity, it's because they are both influenced by the same factors. All right, so we could stop here and chat, or I can very quickly go over another study which will be coming out next week on language endangerment. I think since this might be interesting to people, let's whip through this, but feel free to interrupt at any stage. But I'll just very briefly go over this. So now that we've looked at factors that influence language diversity, we can look at factors that influence language loss. So it's considered about half the languages currently recognised are considered endangered. Now compare that to like amphibians are in an extinction crisis and they actually have lower levels of endangerment than language, and so do mammals and birds. So this really is a crisis that is easily paralleling the biodiversity crisis. And we can also see that language endangerment and loss is non-randomly distributed about the globe. However, when you look at this map of endangered languages, you can actually see that really the hotspots of endangered languages are simply the areas of high language diversity, such as New Guinea. And that's not surprising. If you've got more languages, you expect to have more endangered languages, particularly if they're smaller. So if we plot the proportion of endangered languages, then you get some very interesting patterns emerging. And you can see that some areas are under a much greater threat than others. So this suggests that there are, in addition to the specific things that happen to each individual language, there are also area-wide effects that influence many different languages. And again, we have to control for the fact that areas share a history and an environment, that relatives will tend to be similar in many senses. So we used an endangerment scale based on the EGID scale used in Ethnologue, and I'm happy to talk about that later. And then we chose a bunch of predictive variables that it's hard when you do a global analysis because a lot of the things you really want to know there isn't data on. So we tried to target variables that represent language transmission to children, language shift through socioeconomic change, and also aspects of language policy and environmental change. Look, I'll whip through this pretty quickly, but basically for every language we're looking at its speaker population size, its endangerment level, again, placing it in a landscape. Is it in a hilly country? Are there navigable waterways? Are there many roads that connect up communities? We look at whether there are the major world languages overlaying that language, and aspects of the environment such as growing season, which has been shown to influence diversity. We also want to measure how much the natural environment has been impacted. So for example, through the number of threatened species. the way that the land is used by humans, for example, the increase in urbanisation or the degree to which it's cropland, and then the way that that usage is changing over time. Is there a change in the amount of the built environment and so on? Of course, we include socioeconomic factors such as life expectancy, and we gathered as much information as we could on education, and also describe the language ecology around each language. How many other languages border it? Are there other, what proportion of the speakers in that area speak that language, and what proportion of the languages in the area are endangered? So when we do this, we identify factors that are globally significant, and we identify factors that are significant in particular regions. So for example, pasture and cropland are significant predictors of language endangerment in Africa, but not in Europe. So just looking at the globally significant factors, not surprisingly, the number of first language speakers is the major predictor, but it's not the whole story with language endangerment. You can have languages with a large number of speakers that are endangered because they're not being passed on to children, and you can have languages with a very small number of speakers spoken only in 1 or 2 villages, and yet they're stable because they are being learnt by children. One of the major factors across the world that correlates with language endangerment is road density. Now, we interpret this as meaning it's not just an indicator of economic development, because other indicators of economic development are not significantly associated with language change. Instead, we'd interpret this as a sign of population movement, the connecting up of rural to regional towns, allowing people to move in both directions. What we can show that it's not simply a matter of the more language contact, the more endangered you are, because actually languages that are bordered by more different languages tend to be less endangered rather than more endangered. But the more threatened languages are in the area, the more likely each language is to be threatened. So there's clearly some sort of area-wide impacts on language vitality. And one of these is education. So the more years of schooling, the greater the rate of language endangerment, which is a very worrying result, but it's also echoed by community level studies on endangerment in schooling. Now, I'm running out of time, but I'll just say that because many of these factors change over time, we can also predict language loss into the future. And I'll just very rapidly say that when we do this, we project the likely speaker populations and the landscape and the climate into the future. We get the rather frightening figure that language loss could in fact triple in a very short space of time, and that we could lose over 1,000 languages by the end of the century. And most of these are actually currently poorly documented, and yet most of these languages currently have living first language speakers. So although the situation looks very dire, it's not a lost cause. We still have some time at which to help stabilise language loss and to support communities to revitalise their language. Anyway, I've spoken for far longer than I intended to. So at this point, I'll open it up and say, does anybody want to ask any questions? And we'll also have a quick break before we move on to the next one. But does anybody want to ask anything? I haven't been following the chat, by the way. So if there's anything you put in the chat you want to ask again now.
The chat has been very taken up with words for car.
Oh, excellent. Fantastic. That's very good.
But I'd point everyone to a paper that David Nash has written on words for car in Australian languages if you want to follow up on car words.
Brilliant. I'm so glad to know that even exists. And it would be interesting to see if those kinds of words have a faster or slower rate of change, these sort of neologisms that come into languages. Do they shift faster than other words or not? Yeah. Has anybody got anything they want to ask about the kinds of approaches we're talking about? Now, as I said, Felicity and Jara are going to talk in far more detail about the population level modelling, so hopefully there'll be chances to ask that as we go. And it is very weird having a masterclass where we're all sitting in rooms on our own. It would be so much nicer if we were all sitting around chatting about this over a coffee.
Also weird having a masterclass run by 3 women, but you know, names aside.
I think we can deal with that. We've got some nice beardies on the screen there anyway.
Oh, well, there we go.
So there you go. All right. Do we want to have and by the way, certainly I'm hoping we'll all be able to stick around after the class is finished to chat with anyone. And of course, I'm more than happy for people to contact me by email or whatever to have a chat afterwards if you think of things you want to ask. Shall we have a few minutes break for everyone to grab a coffee and a biscuit and come back at, shall we start again at 5 past? Does that give you enough time, Felicity? Okay, cool.
That sounds good.
All right, so let's all, and I will still be here. So if anybody wants to ask a question while everyone else is chuffing around, finding themselves a snack, a late breakfast. And can I just
hanging out the washing?
Yeah.
Which I just did.
Can I just point out it's even earlier for Felicity. I don't know whether anybody else is, I don't know what time zones people are coming in from. It's about 10 o'clock here in Canberra, so just time for morning coffee. I've brought my own coffee in a thermos, so I'm just about to have that.
Yeah, there's a few Queenslanders there I can see. Good on you.
I hope, is there anybody who's in the middle of the night for them? I don't know whether we've got any international people coming in. Oh, now, while we're here, Felicity, do you want to get your, why don't we start fiddling around for you to put
I'll get my slides up. So if you unshare yours.
Okay, what do I do with that? Stop share.
Yep. And I guess I'll, that's a bit annoying. Oh yeah, I haven't done this on my laptop without a second screen for a while, actually.
Can I just say I'm admiring that beautiful artwork behind you?
Oh, I know, isn't it beautiful? It's Balgo, yeah. So I've forgotten just briefly forgotten the name of the artist. She's an amazing woman. There was some women's songs, Yalu, that were recorded with her and some of the other Balgo women that Miff Turpin and I repatriated a few years ago, and we thought there was a mistake because we put them in iTunes to turn them into MP3s to make them easier to play on different devices, and it was 3 days straight of song recordings. We were sort of weirded out by this because it said 72 hours, and it was 72 hours worth of just straight recordings. Those women could sing, my goodness.
You should just like put that in as a big sesh, like just go straight.
Yeah, that's right.
That would be fantastic. The big Belgian singing bash.
Yeah, that was a disco. Oh my goodness. Now allow Zoom to share your screen. What? This is a new. This is new.
And now, Jo, have you made Felicity a host?
Yes.
It shouldn't make a difference for screen sharing anyway.
Oh, now it wants me to leave the meeting, but I'm not going to do that. Okay, I'll just see if this works now. This is odd. I don't know. Maybe it's because there's been here we go.
Yeah, that's working. Yep.
That works. The annoying thing is, of course, I can't see my notes, but anyway, never mind. I just have to remember what.
Felicity.
Yep.
You should usually be able to right click and click presenter view when you're in full screen, and then it should show you your notes, but us the slides.
Okay, hang on, say that again. Again, slowly, BD.
So start the slideshow, go into full screen like normal.
Yep. So right click.
Yep, and it should have presenter view once you're.
Very nice. And so what can you see though?
We can still see your, oh no, it's showed us your notes now. Sorry, my bad.
Yeah, no, thanks.
It worked on my laptop.
You're a PC user though, right? So.
Yeah, right.
Yeah, maybe, thanks. That's a neat trick for other things, however, I'm sure. So it's quite cold in Canberra.
Yes, it's around 10 degrees yesterday.
Wow,
Can you believe I actually lit the fire yesterday, like in the middle of December, partly to dry out the roof which had leaked, but
wow.
I'm just going to pop out for a second. I'll be back in a minute.
Yep.
Yeah. I also need to refill my water. I'll see you in a minute.
Yeah. Mm. Just trying various tricks, Lindell, but none of them are working. It's all right.
You can try just sharing a portion of your screen. So like you can click and drag the amount of your screen that you want to share.
Oh, okay. But that might not work for presenter view anyway, I think. Oh, I see, but you can then just show the slides. Is that what you're saying?
Yeah.
This might be a bit beyond me. Yeah, I think that's all right. I'm sure I can remember what I was going to say anyway. Thanks, though.
All right, it's about 5 past, so I reckon you could probably start whenever you're ready.
Okay, great.
I'm back.
Yeah, I'll just see if I can. I'll just bring. Sorry about that. It's just going to bring the chat up, but I guess someone else can be monitoring that anyway.
I'll see if I can keep an eye on the chat, and maybe I'll interrupt if. Well, for a start, people should feel free to put their mic on and interrupt anyway. But if there's something that is asked that Jan and I have no idea about, maybe we'll
pipe up
and tell you.
Okay, great.
So you don't need to watch the chat. We'll watch it.
Yeah. Okay. Yeah. Cool. All right, so I'll get started. So I'm zooming in from Yaga and Turrbal country in Brisbane, and I'd pay my respects to the elders here. It's been a real privilege working with Indigenous communities on Indigenous languages over the past 2 decades. And Lindell has already done this, but we're transporting you to Grungy country. And you can see a beautiful picture by a Grungy colleague of mine, Brenda Croft, who's at ANU, of the old fence posts from the original Wave Hill station that was flooded, and in fact flooded by a grungy clever man who was goaded by the station manager, who claimed he didn't have magic powers and couldn't make a flood. So this magic man, Tinker Rudder, created a flood, washed away the station, then they had to move it. But I guess what you can see with this desolate lot of fence posts here is really captures colonisation, and the story that we're telling about language shift and change here is very much about colonisation. So Lindell's already introduced Cassandra Algie, and you saw her in the video before, but I just wanted to acknowledge the work that she's put into this project too. Cassandra and I've worked together for about 18 years, I think. And she's been really instrumental in doing the recordings and that sort of stuff, and the data that underlies this project work. And you'll see various other collaborators as we go along. So the first question I have that I wanted to address for the linguists here is, why do we want to do multivariate analyses in the first place? Because you'll see from what Lindell's been talking about, they're actually an enormous amount of work. And for linguists, this really involves collaborating with people who know a lot more about this stuff than we do. So different kinds of linguistics are really interested in variation in the same way that biologists and evolutionary biologists are as well. So language evolution is driven by variation, where particular variants are selected for over time. And both variationist sociolinguistics and historical linguistics have been tackling the micro and macro ends of language change and variation in different ways. So some of what you saw Lindell talking about is the sort of stuff that historical linguists are interested in, where traditionally they've used the comparative method and now borrow phylogenetic methods from evolutionary biology. Variationist sociolinguistics has traditionally used multivariant analyses to understand the variable use of particular variants of a single variable. I seem to be using the word var quite a lot there. So a nice recent example that I saw, and I've been a bit unaware of, I think because of COVID, is that apparently in Melbourne English, all you Melbourne people out there are losing the distinction between Ellen and Allen and celery and celery. So you say things like Melbourne now, which I find really bizarre. And so that's some work that Deb Lokes has been doing. So variationist sociolinguists have been using things like arboural mixed models and now random forests and sea trees and that sort of thing for smaller data sets. The work that Lindell has been talking about, and Jar will talk about in a great deal more detail, borrows population genetics from the evolutionary biology. And the reason for this is, while it's great being able to study individual variables like that Melbourne example, they are limited in their capacity to map language change and to generalize across different aspects of languages. So when we're using multiple variables, instead we can avoid some of the pitfalls of theorizing on language change based on individual variables. For instance, claims that simplification occurs through language contact. Again, this is something that Lindell touched on. And when we're using multiple variables, they're chosen for their factor variation, which avoids the issue of circular argumentation. So if you're making claims that simplification occurs through language contact and you're picking variables that you notice that are undergoing simplification, you're just going to prove the story that you're wanting to prove. So instead, what we want is an unbiased selection of variables, which avoids what gets called the ascertainment bias that arises from focusing on features that are known to be undergoing specific patterns of change. This is why we don't want to be, for instance, picking variables that we know are undergoing simplification. What we want to do is cast the net really widely and look at variables that are undergoing change and ask of those variables whether simplification, morphological simplification, is driving the selection of those variables. So we've been using Fisher models, and again, and Lindell's already introduced this, so I won't go over it in any more detail, except to say that really this work was the first application of Wright-Fisher models to a large sample of language variants within a single speech community to model broad-scale language change over time. And add a caveat, there was a paper that looked at English, but only looked at about 15 variables, so a much smaller application of it. But there were theoretical models that came before that, sorry, people like, I can't find his name in all of this, but Bill Croft had been working on this sort of stuff theoretically for a while. So back to sociolinguistics and this sort of shift from single to multiple variables. So sociolinguists since Lobov have been looking at the variable use of linguistic features by speakers, and they've been interested in demonstrating how variation in speech communities indexes social categories like the kind of classics of first wave sociolinguistics like age, gender, social class and ethnicity. And most of these analyses have been based on the indexical nature of individual variants against multiple predictors. And these kinds of variants can be things like phonetic, morphosyntactic, and discourse variables. So you have a single variant and a number of predictors which drive whether you use that particular variant of a variable or not. So we've been thinking about what happens when you study language variation across multiple variables and their variants against multiple predictors, and actually why would you want to do this, because it turns out it's quite a lot of work. But you can see generally the model that we're using there. So actually, Lobov actually did think about this, and he noted in back in 1972 that multiple variants often covary. So he, with his study of New York City English, looked at 5 phonetic variables that all separately indexed categories of social status and degrees of formality, and noticed that they actually covaried. And you can think about this, once you start getting variables that are covarying, this is when you start getting sociolects breaking away into dialects, and then presumably this is where new languages emerge from. So this is why we're interested in this. This is why historical linguists as well as sociolinguists are interested in this. This work was then picked up by Guy, who questioned then whether multiple variables cohere in forming sociolects. So he was picking up on this idea that if you have multiple variables that are covarying, then you're going to start to see the emergence of, in this case, sociolects, because we're interested in the relationship between variants and different kinds of social variables. So this is what he called, Gregory Guy called linguistic coherence. So this is an idea that predicts that the usage of clusters of particular variants should be more similar among speakers from different social groups than speakers from different social groups. And it links then individual speakers with sociolects by mapping clusters of variants found in these individuals to sociolect level variation. And since then, there's actually been lots of people who've been working on this problem, because, as I said, it's the thing that perhaps links then individual variation with the creation of sociolects, and then potentially new dialects and languages. So the idea is that we're going from a single variant with multiple predictors to multiple variants with multiple predictors, and creating particular sociolects. We're particularly interested in, when we're thinking about language evolution, in terms of region and age. Of course, age being the snapshot that we take of change over time. The idea being that both time and spatial separation leads to new languages. And just to say, one of the plots that Lindell presented in the previous paper that I think Shah's also going to talk about actually demonstrated coherence in a way that other people haven't been able to do so previously, but using discriminant correspondence analysis, not the population genetics models that we've been using. So I guess I've been answering this question as well. Why do we want to make our lives this difficult? We need new tools. These are sorts of things that verbal or mixed models can't use. We've been using Wright-Fisher models, discriminant correspondence analysis to do this work. What I'm going to be talking about specifically is the fact that you need lots of data across lots of speakers, where lots of speakers have had many opportunities to express particular functions, those functions being the variables that we're interested in. And we want to do this because we want to map variation across an entire language, and we want to be able to select multiple variables for their factor variation rather than a biased selection of individual variables that depending on what our hypotheses are. But while we're doing this, we will end up with a better grasp of how language change occurs. But yeah, there's a lot of work underlying this. And actually, some of what I'll do now is point to some shortcuts that are coming into play that make all of this work a lot more possible and doesn't require 2 decades of field work. Okay, so I'll introduce you a bit more to the Grungy Creole corpus, and it's very nice to have some of the collaborators online, Sasha Vilmont, who was really instrumental in cleaning up this corpus, Bodine Sloan, who also helped transcribe some of this corpus, and I'm not sure if there's some other people I'm missing. But so we're in Gurindji country at the moment. You've seen part of this map before with Lindell. There's a Creole, an English-based Creole language, which is spoken right across Northern Australia. And in some places in Northern Australia, these 2 languages have fused. And so you get mixed languages like Gurindji Creole forming. The other well-known language, of course, is like Warlpiri, which Kamala Shanis has been doing a lot of work on over decades. And we've collaborated on a few interesting aspects of both of these languages. So you've seen this picture before. This is Topsy Dodd. She's one of the senior songs women at Kalkarindji. So I've collaborated a lot with her and Mif Turpin as well on women's songs, and also a song set that Mif has been tracking right across Australia called Wunji Wunji. Wait for a fantastic podcast on this that is coming out on Radio National. So this is Topsy Dodd with her daughter Deborah Vincent and her little daughter Vikara, who's now speaking age. So in this photo, obviously she's not very talkative, but we can say a bit more about the language that she uses now. So Topsy is bilingual, but not just bilingual. She actually speaks other languages too, but the languages we're interested in are Gurindji and Creole. The Creole comes from the cattle stations, whereas Lindell said people were essentially enslaved. They worked, they were forced to work for no pay and then walked off the station in 1966 in protest. So her daughter, Deborah Vincent, has grown up listening to her mother code switching between the languages, between Gurindji and Creole, and there are real patterns in that code switching. There's some other work that I've done on that. But that code switching is essentially fused into a mixed language. So Deborah is the first generation of mixed language speakers, but there's still quite a bit of variable use in that mixed language. Her daughter Vikara is now a speaker of Gurindji Creole as well, but there's much less usage going on in that language. So it's sort of solidifying in many senses. Just thought I'd play some examples. So this is Rosie Smiler, another Gurindji colleague of Cassandra and I, just talking about being jabbed by a catfish, and then her sister's daughter, Jamisha, telling a little story to her 2 little brothers in Gurindji Creole. And you can see some of the picture books that we've used, which were created by Kamala Shanis to create some of this data set. Okay.
Adanya even bushum bari muna dat karungu dan even kun jora dat logo nam Wedding that tree, I think they jump in and I that thing that kajiri and that Dora jump on look at logo. And that guy have been making the jump look at logo. I won't get in them making fire. That's it.
Okay, so it's always nostalgic looking at these videos because those were actually made quite a while ago now. Jamisha now is grown up herself and has children, and one of the things we're hoping to do in the future is actually to record that next generation, so we can give even more time depth to this unfolding story of Grungy Creole. But so if you were just listening to those videos, there might have been some words that you recognize, like in that first section where Rosie was talking about being jabbed, or Samantha being jabbed by a catfish. I don't know if you know, sorry. So catfish have got a spine on them. You have to be very careful when you catch them not to get jabbed because there's poison in the spine and it's extremely painful. It's not dangerous, but just really, really painful, just for those who haven't been catfished before. But anyway, so you would have heard some words like mud, perhaps rubbum, artem from hurt, which are from Creole, and then a whole bunch of words that you wouldn't have understood. And the same with Jamisha's conversation. So you get a sort of sense of the data that we're talking about. So just a bit of an overview of the structure of Gurindji Creole. You get nouns coming from Gurindji here. So we've got Yao, Waran, Waragu, Kaya. We've got a sentence, each one from Rosie and one from Jamisha there. You also get nouns coming from Creole. The only example here is a name, so it sort of doesn't really count, but you get plenty of nouns from Creole as well. You get verbs from Gurindji, so this is originally a co-verb turp for jab, jawara there. You also get verbs coming from Creole, so you've got gone there. And what you in fact have the bin gone jawara is for those who are interested in asymmetrical serial verb construction. You get nominal morphology coming from Gurindji. So you've got that ngu there, which was originally an ergative marker. It's now a nominative marker. Sasha Vilmot and I have had a paper come out in a volume recently about this shift across generations of this particular case marker. You also get the verb phrase structure coming from Creole. So you see the past tense marker bin there, and plus these pronouns, which I suspect are more clitic-like than free forms, but I would like an honour student to work on this. It's a bit of an advertisement if someone's interested. Okay, so generally speaking, we can think of Gurindji Creole, the mixed language that's come about from the previous generation of code switches, has been characterised by the adoption of Gurindji features and Creole features. And note that I'm using this term adoption of. If you think back to Lindell's slides before where we had the balls in a jar, I looked at them and thought of them as lollies, but maybe that's just because it's, I don't know why I'm thinking about lollies Christmas season, but you could see that there was a sampling from jar to jar as you sort of went along. And so Gurindji Creole in a sense is a kind of sampling exercise as well. So you've got some Creole Gurindji features, you've got some Creole features. So, you know, you're selecting some green lollies, some orange lollies. You've also got the development of some innovative features. And also some of the Creole features have been undergoing a kind of acroelectralization process because of increasing contact with English. And I guess, so the question we're asking is, why are some Gurindji features being selected, some Creole features being selected? And we're also interested in these other kinds of innovative features. But here's an example here. This is a dog that's lying under a table. It's part of a director matcher task, which I'll show you shortly. And you can see that there's different ways that Gurindji Creole speakers have of expressing this. So if you look at that first sentence there, you've got quite acrolectal features. So you've got big one, which is a little bit more English-like. You've got next to being used instead of one side here, which is the more basilectal Creole form. And you've got bigger one, which is the more basilectal form as well, which comes from biggest, but isn't the superlative form from English. It's just the adjective. But you can see a lot of features that all have in common. So everybody's using the word for dog, wallagool, everybody's using the Creole past tense marker, the grungy coverb muggen, and then you get innovative features too. So for some speakers, you might get just the grungy locative marker being used. For other speakers, you get this double marked form where you get the Creole preposition langa, and you get it double marked with the grungy locative marker there as well. So this is what we're calling innovative elements. There's different kinds of innovative elements. This is one kind where you get double marking. So this is kind of some of the data that's underlying our database. So we're interested in, when people are speaking Gurindji Creole, they're listening to previous generations, what's shaping the language change? Why are they sampling sometimes Gurindji features, sometimes Creole features? Are there patterns of language shift that are predictable? Are there environmental features or sociolinguistic factors which increase language endangerment? What are the factors that help preserve heritage languages like Gurindji? And are all language variants equally prone to loss in language contact, or are some more resistant to loss? So, for instance, is production lost faster than comprehension? This is claimed in the literature. Is grammar more susceptible to language change than lexicon? Again, this is claimed in the literature. Are nouns more easily adopted than verbs? Again, claims made in the literature that we can test. And I'm not going to talk about those. I think Jar will. Anyway, so what we've done is developed a multivariant corpus. So if we go back to our example of the dog sleeping under the table and one of these particular features, so this is the spatial static location feature. You can see that you can express this in this community by either using just a Gurindji case marker, the locative marker. You could also just use a Creole preposition, or you could use the innovative double marked construction there. So some speakers might have pattern 1 where they're only using the Gurindji case marker. They never use the preposition or the double marking strategy. Some speakers might use pattern 2 where they've got the Creole preposition, but not using any other strategies. Pattern 3, they might be using the Gurindji case marker in some cases where they're expressing static location, like the dog sleeping under the table. Other times they might use langa, the preposition. And then the other pattern that we found in the corpus was some speakers use just the preposition, and in other cases they use this double marking case. So this is just one variable. Static location is just one variable with 3 different variants and 4 different patterns of use amongst speakers. We then did this using 174 variables and 346 variants. And I know that Sasha Vilmot is in the audience. I'm not sure if there were any of the other wonderful, wonderful summer research students who actually helped extract this data, but it was well, it was kind of a fun 3 months of summer doing this. Okay, so what we then came up with was what Linda was talking about, much nicer slides actually, was this idea of a lingua type. So the lingua type is an individual speaker's pattern of variance sampled across 174 variables. So these are the patterns. So it's 174 variables and the kinds of patterns that they're using across all these variables. So it's essentially, this idea of a lingua type is essentially an unbiased sample of a person's idiolect. So the idiolect is their entire speech and the variables and variants that get used in their speech. The lingua type is a sample of these. And it's analogous, as Linda was talking about, to an individual's genotype. So, so here we have our static location here with the potential variants and the different kinds of patterns that an individual speaker might use. So a speaker is going to have one of these patterns, and then we do this across 174 variables. So this is all derived from a corpus which is just about to go live in Paradisac. It's the largest Australian language corpus, a corpus of an Australian language, sorry. So it's 165 hours that have been transcribed, translated, anonymised and annotated across 157 speakers. And this represents about 500,000 words. I was in, even though speakers like to compare their corpora and how big their corpora is, you know, the usual kinds of pissing competitions that different disciplines like to have according to the number of words that they have. It doesn't work very well in linguistics. So this is a similar sized corpus actually to the Australian English corpus that was being talked about yesterday by Catherine Travis and her wonderful team. And that represents more like 1 million words. But of course, English has much less morphology than Grungy, so in Grungy Creole. So you're going to have a corpus that has half the number of words, but an awful lot more morphology. So this sort of word count for a corpus thing doesn't work very well, just as a bit of an off topic. So the data set then that was drawn from this is a binary coded data set. So Jar will talk a bit more about whether we want binary coding or there's better ways of doing this. And it's a subset of people. We've got 78 people for whom they've expressed these 174 variants, so we could come up with a nice data set with them. Okay, so I'm going to then backtrack a little bit. That's the data set. How did we collect the field data in the first place? So when we initially were doing this, I had never met Jar, I had never met Lindell. I wasn't collecting data to create a multivariate corpus. If I were to do this now, I'd do it slightly differently and much quicker. And I'll talk about some of the sort of innovations that have been happening within the Centre of Excellence within CoEDL to speed up this process a bit more so that you too don't need to spend 20 years of your life doing this. Okay, so what we need are multivariate corpora. And these are basically comparable variables that have been recorded across multiple speakers from different generations and preferably different regions. Now, the Grungy Creole dataset does not have different regions involved. We've got a Shawi dataset that we've been working with Lewis Miguel, which has different regions as well. So Shana Poplak would tell us that conversational data is the gold standard, and she's done amazing work in Toronto collecting French, English, code switching data, but you know what, that stuff is so much easier to transcribe and it's easier to get conversational data. Also, if you're only going to collect conversational data, you need much, much more of it. So conversational data and free narrative does provide the basis for us for deciding on the variables to target, but it's not comparable enough to build a data set for analysis. And that's because when people are having conversations and telling free narratives, the topics are free ranging. You can't guarantee that people are going to use particular lexical items and grammatical constructions. So it's really hard to develop something where you've got comparable data sets across speakers, unless you've just got hours and hours and hours, hundreds of hours of data. So this might be possible if your variables are phonetic. So if you're a phonetician, you're interested in phonetic variables, this kind of data set might work for you. So what we did instead was we supplemented the data using semi-formal elicitation methods. So I'll show you some examples of these: director matching tasks, picture prompt narratives. And the point of this was to ensure that speakers have the opportunity to express lexical, phonetic, and morphosyntactic features using a variant of choice. Okay, so we need data sets, essentially. The characteristics of a multivariate data set is that there needs to be sufficient variation. And you might get these from different places. So you might find a well-documented data set of language change through historical corpora. For instance, English is obvious. So you might have fewer variables, but potentially more speakers and regional variation. You need that regional variation to really start narrowing into language evolution. And you should be able to then use frequency data if you've got larger data sets, whereas this data set's built off binaries. You could also target features that change rapidly, so phonetic features again. So again, you might have fewer variables, but potentially more speakers and regional variation. And again, you should be able to get frequency based data. The other place that you can look for to produce really good multivariate databases are situations of contact-induced language change. And this is what we're talking about here. So grungy Creole, there's rapid language change, rampant variation. It means that we've got lots of variables, potentially fewer speakers because these are small speech communities. This is actually, I've forgotten the number, Lindell, you might remember. I think it's a sample of 15% of the speech community, which is actually a very large percentage of the speech community that's been sampled, even though it's 78 speakers. But we've got lots of variables, fewer speakers, but a large percentage of the speaker population. We don't have regional variation in this. This is something that we need. And this data set uses binary data, which we've done so that we could use it with rarer variants, so variants that don't crop up in the data set so much. Okay, so Jar might talk about this. I don't know if you want to say something about this now, actually, Jar, but continuous data is better than binary data. Did you want to say something about this now, Jar?
So the usage frequency, observe the usage frequency. So for example, how many times this person used this variance in the example would be more informative than whether this person used this variant in the sample. So ideally, you want to count how many times you've given this person a chance to use a particular variable and how many times this person used each of the variants. That's the most informative data. Yeah.
Yeah, great, thanks. And so correct me if I'm wrong, but also the zeros then in the data set need to be informative, that rather than missing data points, but that you have fixes for this sort of stuff too, right?
Yeah, so if you're the total number of time you allow the user to use a particular variable equals to 0, which means it has all zeros in all variants, this data is the least informative data. As long as you have one chance for this person to use a particular variable and this person choose a variant to use, then it will be way more informative than just all zeros for all variants. But the most informative one is you allow the person multiple times to use a variable, and this person gives you, and you count how many times this person used different variants.
Yeah, great. So you end up with this data set, which was started at the initiation of it was actually through this summer research program. So we've got a number of participants. So these are all the participants here. As you can see, they've been anonymized, they've been given numbers. They come from 3 different generations. So third generation are the kids, second generation, the adults, first generation are the older adults, the grandparent generation. And these are just 2 variables here. So the use of the ablative, and the expression of the ablative and the expression of the dative. So these are case markers, and they get a particular feature number. So for instance, you might express the ablative using Brom or Budum here. So you've got Budum, which is the really basilectal Creole variant. You've got Brom, which is the lighter variant, and then you've got 2 grungy variants which you can't see fully here. One is ngini and the other one is ngini, so it's got a velar nasal on the end of it. And so you can see that this participant, person number 54, who's of the younger generation, only used when they were expressing the ablative, so sort of expressions of from somewhere, only used the Brom, the lighter variant. If you look at someone, for instance, from generation 2 here, P26, person 26, this person uses that lighter Brom as well, but they also use the grungy ngini and ngini with the velar as well. And then if you look at someone, and I've got these variants the other way around, but it doesn't matter. Someone who's from the grandparent variation, grandparent generation, they're not using the Creole variant at all. They're not using the nginin with the nasal end at all. They're just using the ngini variant here. And we can say the same about the dative as well. So we've got a Creole form blunganda evens from belonger. We've got bor, which is the lighter form, and then the dative marker. And again, each participant is marked for their pattern. Essentially, this is their pattern as to whether they're using these forms or not. And again, this was done on binary data. So this is just ones and zeros, but what you would really want to be doing is saying, well, yeah, sure, they use bor to express the dative for, and they also use the grungy form, a dative marker, but actually, is this really 50%, or are they using bor more frequently than they're using the dative? And if you've got that kind of data, you can start asking more questions of the data set, but we're just using a binary data set. Okay, so I'll just talk about some of the actual the data elicitation methods that we were using. So a lot of this comes from director-matcher tasks. These come out of semantic typological research, like those spatial relations, reciprocal studies that Steve Levinson's group at the NPI Nijmegen created. And we use this method to produce comparable data across speakers within a single language rather than across, whoops, I've lost my screen here, across languages, which is what semantic typology does. We've used what I've dubbed a peer elicitation approach, and this is to ensure that the data captured is as close to everyday speech as possible. So I talk a bit about this in this textbook that was written with Jenny Green and Miff Turpin. And this is something that Chana Poplak also talks quite a lot about when you're doing sociolinguistic interviews, for instance, is that you don't want outsiders like myself to be the primary interlocutor, because if you do so, then that person is accommodating to you, and the variants that are popping up in their speech aren't the variants that they'd normally be using. They're accommodated variants to the outsider that they're talking to. And this is where someone like Cassandra Algie and other Grungy women who I've worked with are really crucial. So using a local field assistant like Cassandra Algie replaces that linguist speaker dyad, which is really common in linguistic research, with a peer-peer dyad. And as I was saying, it's ensuring that the data is not skewed towards a Gurindji person of another generation, or for instance, an English-speaking researcher. And actually, a lot of the problems that I had were that people would be using Gurindji Creole with me, but they'd be using the really traditional Gurindji case allomorphy, for instance, rather than the ones that they use within their own peer group, because then I kind of spend a lot of time with the older people. So a weird kind of accommodation, but an accommodation effect nonetheless. Nice that I'm associated with old people. Okay, so here's just a photo of a director-matcher task, and then I'll show you a little video of Cassandra doing them. So here we've got Cassandra on the video camera, me just looking interested, but I've actually got a notebook there. And then you've got Jamisha Smiler, who you met before, with her, who's the director. So she's the one who's describing the pictures on the board, and then the matcher, Kuria there, who's got a set of pictures in front of her, and she's listening to Jamisha and then picking the right picture. And the idea is that all of these pictures just differ minimally. So if you look down here, for instance, you can see a woman whispering to a girl, to a man, and to another woman. And all you linguists out there, you guessed it, what I'm after is some kind of dative construction here, whether it's a Creole preposition or a Gurindji dative case marker. So that's what we're looking for. So I'll just give you an example of Cassandra actually undertaking this task, and you'll see the dog on the table again.
Pero wan kajari tingna kara bo yat karo mengepta. Wanga. Wan malo magen table kanjara. Tet mama ando mdukam nanga bo wan karo.
Okay, so that example there, we've got our dog under the table again. So again, remember you can be using the case marker, the preposition, or the double marking. I forgot to check which one Cassandra's using. I think she was using this double marked construction. So she said langa table. And again, this is across 174 speakers. So just one of the ways that this has now been sped up is Lewis McGill was doing this research also in Balgo in Northern Australia. We've now got ways of doing this using iPads so that the utterances are recorded directly onto iPads now, which takes out some of the clunkiness of doing all of this manually, which you've seen with, laminated boards and pictures, and then recordings, and that whole workflow of extracting recordings from the devices, putting them onto your computer. And I have to say, for all you young ones out there, I was doing this with tape recorders and digital videotape, which meant that when we were digitizing recordings that actually happen in real time too. So you have no idea already how much easier your life is than my life 20 years ago. But anyway, now this is even easier because we can do these kinds of direct-to-matcher tasks on iPads, which means that the recordings are already there and already being ingested into things like Elan, although it's Clan that I've used for these kinds of tasks. Here's some other examples. So these are the man and tree games, which some of you might know from again, Steve Levinson's group. So you've got people who've got minimally different pictures of trees, men standing beside trees. These all look really boring, but everybody loves doing these tasks actually at Grungy. So you've got a director. So this is Amanda Vincent here, who's got a set of a board in front of her with the Velcroed pictures on it. Little tips, by the way, if you are doing this stuff manually and not with iPads, make sure that the Velcro that you use for the individual pictures is the fluffy side, not the hook side, or you'll end up with all of the debris in the community that you're working on the back of your pictures, and they will no longer stick to the board. Just a small little tip. I'm sure you're all using iPads now. But anyway, so you've got Amanda Vincent, you've got Thelma Smiler there, who's got the loose pictures in front of her. You can see the board between them. In this picture, you've got Aliana Smiler, who's the director, and then Conway there, who's got the pictures in front of him. They can't see what the other person's saying. They describe the picture, in the case of the adults, mostly using north, south, east, and west. In the case of the kids, sometimes using cardinals, sometimes using a sort of pseudo-cardinal place names, which is very interesting paper. If you're interested in this, Vivian Dunn did her honours thesis on this work. Okay, so these are some more direct-to-matcher tasks. We also had tasks that were gamified and appropriate for the community. So Grungy and lots of communities in Australia are big card game places. So we made these kinds of card games. In this case, we've got head and tail. The kids were asking and the adults were asking, do you have the head of the turtle, the tail of the turtle, or whatever? The idea of this is to look at inalienable possessive constructions to see what kinds of case markers, if any, people were using. And again, you get huge amounts of data on this very, very quickly just through these kinds of games. And the kids all really enjoy doing it. We have Cassandra, again, by the way, I should point out, she's often got an Essendon beanie on with all of these tasks. And the other kind of data that we have a lot of picture prompt narratives. So this is a classic one, the frog stories, which are used right across the world. They've been used for cross-linguistic tasks for a very long time, since I think the 70s. So this is Jovi Algie, who's describing one of the frog stories. This is, a picture from the frog stories. You can see there's no words, so he has to make up the story for it. This means that we get lots of instances of the word dog, the word frog, nakman or cane toad, the word boy, sometimes malju, sometimes karu. And lots of, it means that we get lots of speakers describing the same pictures, so that we have lots of variants of these particular variables You can see Cassandra recording Jovi telling the story. So some of you, we've produced lots of papers from these data sets. Bodine Sloan, who I see online, produced a very nice honours thesis last year and gave a talk at the Australian Linguistic Society conference just this week, looking just at the nouns and verbs across generations to see whether there were changes in the use of grungy and creole across generations and as the kids aged. Okay, so frog stories, there are actually 4 frog story books, not just the one classic one. So we used all of those. We've also got Carmelo Shaughnessy's monster story set. So I think there's about 6 of those as well. And the kids really enjoyed those books, and the adults were very happy to do them as well. We also had quite a lot of comprehension data in the story. God, I just realised it's 10 to 10 already. I've been diverting a bit. So this is an example of a Peabody picture vocabulary task where, for instance, somebody, so this is Keith and Barry here, and Cassandra's again running the task, note the Essendon beanie, would hear the word, the Gurindji word Gidoa for goanna, and then would choose which one. So these are all smaller data sets that have had individual papers published on them, but all then became part of the large data set. So just quickly to acknowledge all of the amazing teams that transcribe this data through a number of research programs here. Some of you I can see online. Amazing work. So students who'd never heard Gurindji Creole before, but became fantastic Gurindji Creole transcribers and then annotating these data sets. As you can see, enormous amounts of annotation and transcription. Sasha Vilmont, I particularly want to thank. She was the person who went through the data sets and cleaned them up so that we could do these analyses really quickly and cleanly and pull out examples of these. So just to show you how they're coded, it's the annotation that's important. So we've got part of speech. The nouns are all coded for animacy, for the language that they come from, for morphological category. Here's a past tense marker. Verbs are all annotated for transitivity. The lexeme, obviously, dog, so you can do searches for dog and then see whether you're using a Gurindji or Creole variant. And grammatical relation to subject markers. And that sort of stuff was very hard to code because basically you're trying to code every single noun for their grammatical relation. And Sasha came up with some absolutely clever shortcuts so that a lot of this work could be done, could be automated using Python scripts, but then we did a lot of sort of bits of manual cleanup as well. So fantastic work. So I'll just finish by saying that data set, the Gurindji Creole data set, wonderful as it is binary. It's just intergenerational data. What we want to be doing is adding regional data. So the idea would be that you have, this is essentially like the Gurindji Creole data set where you've got 3 generations, a whole bag of variants in region 1. The idea then being that that group of people then go to a number of different places, and we have this with the Shawi data, and so we then get 3 different kinds of regions. Generation 3 becomes generation 1, so they're taking that same bag of variants with them. Lindell was talking a little bit about this with those island examples, for instance. But then that generation then has children. They carry over some of those variants, but in other places they pick different kinds of variants. So again, think about those lolly jars. We have a 3rd generation then where again you've got different variants being picked across the generations. So this is what we want then, is to be trying to capture regional data as well, so we can get a bit closer to modelling exactly how language evolution works. But I think I'm going to yeah, maybe I won't go through the Shawi data set because it might be a bit too quick and too much. I'd rather maybe take some questions from people. So I might stop sharing this so that we're currently writing up that Shawi data for anyone who's interested in it. But if anyone has questions about the field methods and the creation of the data sets that I can answer. It looks like Jar has been answering a lot of these through the chat that I can now see.
Hi, yeah, I have a question about, could you clarify more about the reason that the data is currently binary and what's the potential for using continuous data? Because I do wonder, like, in this situation of really radical rapid shift, you can see a clear split where people never use particular variants, but that wouldn't be the case in most other language situations.
Yeah, so I'll start by answering that, then Jean might have something to say. I imagine one of the reasons you're asking this, Sasha, is that when you mob all were extracting the data, it was actually frequency-based data. You counted all of the, you probably remember this, I'm sure it was quite painful. Good job. The reason that we ended up reducing it to binary data is that there are some variants that come up or variables, sorry, that are really frequent. Things like articles, for instance, like that sort of thing. But there are other variants that are really rare. And if we kept the frequency data, we would have had to have chuck out all of the rare variants because they only cropped up a couple of times. So that's why we ended up reducing the whole data set to frequency data. But Jar will tell you now that you can actually have mixed data sets where you can have both frequency and binary data. So over to Jar.
So it is totally from a mathematical point of view. So if you know how many times you're giving this person the chance to use the variable, and then the number of times this person using a particular variant would follow some distribution called a multinomial distribution. So it's just easy to mathematically model it. If only we have a binary or data, whether the person use it or not, we don't actually know how often this person use. So all we can do is, okay, for example, in this n number of chances that the person can use the variable, it could be like one time the person used this particular variable, it could be n-1 time this person used the variable. So we need to integrate over all this possibility, and that's lose a bit of information on how often this person used this particular variant. You can still model it, it's just losing all these useful information. And because Wright Fisher model predicts the expected usage frequency of a variant of a speaker, so the usage frequency itself would be a very variable information.
Yeah, the reason I did ask is because like the variables in Python data that are really associated with young people, so older people do actually still use them occasionally as much as they don't want to admit it. Yeah. So, you know, that wouldn't show up for as being any difference between generations, even though there is.
Yeah, exactly, because then you just have ones and zeros and it would be showing older people using them, even if they're using them far less frequently. So what you want is that frequency data. Yeah.
I think it's also worth emphasizing here the importance of what we call a true zero. So when you saw Felicity's data and they're coded for ones and zeros, one thing that's important about the way the data was collected is if you just have free conversational narrative and then someone doesn't use the grungy word for frog, is that because they just never got around to talking about frogs? Or whereas with the story, the picture prompt things, they've been given lots of different chances to say frog, so you're going to find out whether they use the grungy version, the creole version, or both, or potentially neither. And this makes a difference because if you have gaps in your data set where you just don't know whether someone's really been motivated to use the word frog or not, then that zero is non-informative. It doesn't tell you whether that person has either variant in their personal language makeup. So it seems like a trivial point, but having true zeros where someone's been given the chance to use something and they just haven't done it is a really important difference from having not recorded them using it.
Yeah.
Sorry, I hope that makes some kind of sense.
Yeah, thanks.
Yeah, absolutely. And that's why I spent quite a lot of time talking about these multivariant databases and the corpora that I'm talking about. This is this is actually a new kind of corpora that linguists have been using, like with this semantic typology and that sort of stuff, for very specific purposes, looking at language variation cross-linguistically across languages, but this kind of multivariant corpora is quite new to sociolinguistics and language evolution. So this is why I was spending quite a lot of time talking about how we create them. And the idea of this corpora is that you're forcing people to express particular variables in different ways so that you get real zeros in the data sets. You could create a multivariate corpora on conversational data alone, but you would need hundreds and hundreds of hours of recordings to make that happen. And quite frankly, it's not going to. So you are sacrificing some sort of naturalness in creating these corpora. The other thing I meant to say actually is that Ben Foley, who a few of you might know, has recently implemented, it's called WAV2, and I've forgotten what's it?
To VEC?
Ah, great, thanks, WAV2VEC, thanks, Sasha. So he's just implemented this in ELPIS, which is a fabulous transcription tool. It's getting something like 90% accuracy in transcribing Grungy Creole. So what he's doing is he's comparing Grungy Creole. Yeah, it's bloody amazing. For anyone who doesn't know anything about transcription takes, for every hour of data, it takes about 10 hours to transcribe. And that's not even annotating and all the rest of it. It's enormous amounts of work. What Ben has been doing within the Centre of Excellence is he's now creating ways where this transcription is done automatically and instantly, so that when we get to trying to create these multivariant databases, we're not going to be spending years creating them. The time will be spent in recording really good data, and then the transcription will be done automatically, and a lot of the coding is already automated. So this is an absolute game changer, and we're going to start seeing some really fantastic data sets emerging from this, I think. So good on you, Ben Foley. It's phenomenal work.
Yeah, awesome. Yeah. Thanks.
This is, by the way, this has just happened in the last couple of weeks, so we're really excited by it. And he was going to be presenting a masterclass on this afternoon, is unfortunately sick, so I can't even give you the excited, you must go and see Ben's masterclass to change your life if you're a field linguist forever.
There's a question in the chat that I don't know what it means.
Which?
Wave to back.
Yeah, sorry, I could find it. I'm just trying to think if I can find it quickly. Yep, maybe while somebody else say something and I'll see if I can find this quickly.
Well, maybe it would be a good chance to have a quick break and come back for Jar at if we start at 10 past, does that give you enough time?
Yeah, of course. Yeah.
Okay, cool. So how about anyone's obviously absolutely free to stay and chat, either in the chat box or by microphone, but you're also free to pop out for a quick break. And if Jar starts at 10 past 11, that gives time for people to go and grab a sandwich or whatever they want to do. And we'll see you back. I've got to work out how to get the heater to go back on.
And there's a bit of advertising for Ben's WAV2VEC2 in ELPIS that I've just put in chat that I just found quickly. Get warm, Lindell, get warm.
Yeah, cool. All right, see you all back at 10 past 11.
Felicity, that means we should write our paper soon.
Yes, absolutely. When are you finishing a PhD again?
Hopefully mid-year.
Oh, wow, really? Fantastic. Okay. Yep. Yep. And I'll talk to you a little bit about this other project that I emailed you about as well, because this is some of what I want to be doing in that project. If you know, you haven't got bigger and better things that you're doing.
Yeah, it's awesome stuff.
Yeah. Yeah, so Ben's also working with Pinterby literature data sets from Purple House, so the dialysis mob. Which, you know, accuracy is really important in those situations because it's life or death, whereas, you know, Grungy Creole data sets less so. So he's very, you know, he's very mindful of that. So that's a Western Desert data set, and he's just getting amazing results with this. So
so cool.
Yeah,
amazing to see it all come together after so many years.
Yeah, well, that sort of centre of excellence buys you as years. Very hard to do with small grants. I had a text message from Ben before. He's had a vaccination reaction, so he's just knocked out. I assume COVID vaccination reaction, so they'll be rescheduling it. So I guess stay tuned for Ben's presentation on this stuff. He was very excited to be presenting it because it's really just been the last couple of weeks that they've been able to implement the WAV to VEC2.
Awesome.
Yeah.
While we're waiting, could I'd be interested to hear if anyone's kind of taken the work you're doing and used it for especially like acoustic analysis and phonetic analysis, or if there's any scope for that and how it would kind of like need to be altered for that.
So we've been using it on a Shawi data set from South America, Peru. There are some phonetic variables in that one. So basically, you do it in the same way, except with the phonetic variables, you could be using, it depends on what you're doing, but you can be using measures, I guess, of formant frequencies or frication spikes. It depends on what you're interested in, but then you would be using frequency data rather than binary data as well. You could actually be using binary data if you're looking at but the frequency data would be more interesting. So for instance, if you're looking at frication and I guess, continuous data between sort of fricative stop variation, that sort of thing. So, yeah. But yeah, no, I mean, phonetic data is great. I'm not a phonetician, so it's not work that I've done, but phonetic data is great because you can get lots of data points out of much smaller data sets, because unless it's a really rare phoneme, or you've got a language that has a gazillion different phonemes, then those phonemes, which are your variables, are going to have lots of phonetic instantiations of them, which are your variants. So yeah, I think using phonetic data would be great. And not hard, yeah.
Yeah, has anyone looked at any like phonetics and acoustics with your grungy creole stuff?
Yeah, so we've got a couple of papers. So we've looked at stop fricative variation. We've got actually, we have a fantastic data set that's got about 8,000 data points on it that is mostly coded up, that we would love an honest student to work on. So this is Eric Round and I, so I'm not sure where you're at, but we've got a really nice project that's set up and ready to go. But we've looked at stop fricative comprehension data, so perception. We've looked at stop voicing perception as well. We've got production papers on those. This one paper that we'd love an honest student to work on is production data that again comes out of the Grungy Creole corpus. So we've got a few different papers, and I've worked a bit with Carolyn Jones on baby talk. In Grungy it gets called Janyalup. So the different sorts of phonetic sort of fun things that people do, like there's a lot of turning things into palatals, which is really cutesy in Gurindji. That's cool. So I've done that sort of work, but yeah, there's plenty of scope for phonetic work.
Thank you. That sounds so exciting.
And I have to say, I'm dreadful with phonetic work now, because we had a wonderful student a number of years ago who did his honours, Tom Enover, on hard Gurindji data. So it's just hard Gurindji data listening to stops, and he kept telling me that I transcribed stops where there weren't stops. This was often in things like ergative markings. So I was writing ngu where he was saying that the speaker is really just saying ngu. So I now have a kind of phonetic ear, a phonemic ear, not a phonetic ear. And I think what these data sets really need are newbies who don't know the language who have really good ears. So I'm hopeless at phonetic studies now, because I hear things that actually aren't there because I just transcribe everything phonetically.
That's the classic problem when you start getting decent with the language, yeah.
Yeah, exactly. Yeah.
Awesome. Thank you so much.
Thanks for your question.
Okay. We're about 11 now. So if everyone's back from their quick break, Bodine, I've just seen there's a question from you. How about if I'll come back to that at the end. After Zha's talk, we'll have a general discussion about any of the questions raised. And I realise that some of you might need to run on to other things afterwards, but anyone who is able to stick around, then we can stay around and chat. But how about for now, we move on with Zha's description of the method. So Zha's done amazing work in adapting evolutionary methods to, so in other words, we don't take evolutionary methods straight off the shelf and just plug linguistic data into them. We always have to be cognizant of the differences, both in the way the data is coded, but also in the way that language evolves. And it's important that we are always think very carefully about what parts of the methods are appropriate to language data and what parts need to be changed. And this is where Zha has done some absolutely amazing work, which hopefully we can hear all about now. And then at the end of that, we can have both questions specific to Zha's work and also just some general questions. So Bodine, make sure you prompt me again to answer your question at the end. Okay, cool. Thanks, Zha.
Yeah, just to make sure that we save Bodine's questions about the matrices for species endangerment and linguistic endangerment. We will answer this after my talk. Don't worry. In today's class, the first section, Lindell gave us an overview on what kind of evolutionary model we can use to study language change on population level. And then the second part, Felicity gave us an overview on what is the best way to collect informative data and to study language change on population level using evolutionary approach. So in the third section of this class, I'm going to talk a little bit about the theory behind why and how we can apply Wright-Fisher models. Or here, I also going to introduce a model called Morass model, which is mathematically equivalent to Wright-Fisher models under some limits. The reason that I want to introduce this model here, because I think it would be more intuitive for you to see the connection between the random sampling process and the memory update of our language usage. And the second part of my talk, I'm going to give you a short demonstration of how to apply those multidimensional statistical analysis that Felicity and Lindell talk about in their talks in R. So if you are familiar in R, that would be great. If you're not, but don't worry, R language is a very kind of straightforward language to learn. And all the code and the grungy creole datasets are available on my GitHub site. So it's in this site. And the grungy creole dataset was provided as a practice dataset for you to apply those methods. Of course, those methods is generalized enough that you can also apply them to your own dataset and answer your own questions. And in particular, there might be 3 repositories that you would be interested in looking at. The first is called Bayes' VAR rule. This is the new method that I recently developed. The paper is still under review, and hopefully you will be able to read the paper very soon. Basically, I applied a Bayesian framework to incorporate the Wright-Fisher model as the noun models into multivariate regression that Lindell and Felicity have talked about in their talk. And also the concept is similar to the variable rule analysis in social linguistics. So that's why I called it a Bayesian variable rule analysis, just to give my salute to above. And the second repository that you may want to check is that is testing language coherence. These are the codes and the data that we use to plot that beautiful colorful plot where we have both speakers and variants on the same space. So you can test for linguistic coherence using those methods. And I'm going to talk a little bit about how we apply this criminal corresponding analysis to visualize those multidimensional data in language. And the last repository is the code we use to conduct this language endangerment case study. The paper will become available very soon. And the The code include our methods to account for spatial and phylogenetic autocorrelation. So if you are interested, you should check it, but I'm not going to touch it in this talk because I want to focus the talk on population level analysis rather than language level analysis. So that's the overview of the talk schedule, and let's start. The first thing I want to do is to introduce the Wright-Fisher model. Lindell has talked a little bit about this, so I want to focus more on the explicit link between the randomly sampling process and how we update our usage frequency of a variant of a language variable based on the conversation that we have heard. So let's consider there are two variants here of a variable. For example, the variable can be the meaning of dog, and we have big A could be a grungy word for dog, little a could be a creole word for dog, just with an example. And at time t, this is our memory. Our memory has 5 occurrences that we have heard a grungy, a big A variants have been used in the previous conversations, and 3 occurrences of little a variants have been used in the conversation. So, and then we heard a new conversation. Assuming that our speaker community is well mixing, so everybody freely and uniformly interacting with each other, which is obviously not a very reasonable assumption. So, but don't worry, in the future slides, I'm going to explain how we can relax this assumption. But for now, just assume that we are in a perfectly mixing community where everybody speaks freely with each other. And so the usage frequency of variants in the whole community would resemble the usage frequency that we remember in our memory. Okay. And then based on this new token that we have heard in a new conversation, we would update our memory. We would update the tokens we stored in our memory. And this is essentially similar by randomly sampling variants in our previous memory to update our new memory. And turns out the probability of any memory configuration in the next time step when we update it has a very beautiful mathematical rule, and because it just follows a binomial distribution where the parameter in the distribution is only determined by the number of big A we have in the memory in the previous time step. Okay. So that's the basic idea of Wright-Fisher and the basic link between this randomly sampling and how we can update language usage in our memory. Moray's model is a sister model in Wright-Fisher models. And the reason for that is because those two models would have the same mathematical formula when we approximate those models as a continuous time process. Mathematicians call those models in a continuous process as a Wright-Fisher process. So Wright-Fisher process is the model that we are going to use to model updates in language usage in a community. Okay. And Moray's model may be more intuitive for you to see the link between the randomly sampling process and the memory update. So again, we have 2 variants of a variable, big A and little a. Initially, our memory, we remember one big A was used in previous conversation and 4 little a. Okay, and then we have a new conversation and we heard a big A was used in that conversation. So how we update our memory, we randomly select the slots of little a in our memory. For example, we select this little a, and then we replace this little a with big A. Okay, so that's the basic idea of Moray's model, and I hope it's a more intuitive way for you to understand the link. Okay. So the applying Wright-Fisher process to model language change in a community is not a new idea. I think the idea was first proposed by this paper, Baxter, in 2008. Yeah, and later on, there are the first model I think is in 2000. It's called exemplar theory. It was proposed firstly in psychology and then borrowed by linguistics to model word frequency in our memory. The main differences between the exemplar theory and the Wright-Fisher process is exemplar theory has an extra layer. So it first explains how we store a continuous sound into distinct words in our memory. Well, Wright-Fisher process starts with assuming that different words are distinct enough, so we always store them as distinct words in our memory. So, and the next layer would just be random sampling. So exemplar theory would be kind of a multi-layer example where the underlayer is Wright-Fisher process. I hope that's clearer. And because of that, the exemplar theory has no analytical solution. And so we have to use simulations to approximate the probability distribution. But if your data have included words that have distinct sounds, so you don't need to worry about this extra layer, you can just apply the Wright-Fisher process. And the nice thing about Wright-Fisher process is that it has a beautiful analytical solution, which is a direct distribution, as I will talk about later. And other paper like this one and that one, they have proposed a similar idea of how to apply Wright-Fisher process to predict the usage frequency of a speaker in the community. Another paper is by these people. They actually apply Bayesian learning. The similar thing that I talked about before, how we update memory based on how many times a variant was used in the previous conversation that we have heard. And the nice thing about this paper is that they actually show when we have intrinsic usage bias to use a particular variant, the solution is actually converged to the same solution for Wright-Fisher process with mutation. And I will talk about this in the next slides. So in these slides, I just basically gives you a nice visualization of how we apply Wright-Fisher process into the update of our usage frequency of a variant. So imagine that this is my brain, and English is not my native language. So the first time I came to Australia, I have no idea that how people call, what kind of words people use for soda. And the only thing I know that there are 3 variants: soda, soft drink, or fizzy drink. But I don't know which word that the local people would prefer to use. So when I just came to Australia, my initial usage frequency, which is theta 0, would be uniform for all those 3 variants. Okay, I'm also constrained by some memory limits, that is a big N parameter. And now I arrived in Australia and I collect all these conversations around me. I heard 4 incidents that people use soda, and 2 people use soft drink, 2 people using fizzy drink. So this is the data I have gained from the environment, and that's called X0. And then based on the data, I want to update my language usage so that it's easier for me to communicate with the local people. And of course, based on X0, the data I collected from the conversation, I would start to use soda more too. And then I will generate new tokens to the conversation in the community. So I produce more sodas to the community, and now the frequency of soda in the community will increase over time. And the X1 is the new data I input into the community. So this is the basic idea of how we update our memories and contribute to the usage frequency of a variant in the whole community. And if we iterate this process for n number of times, then we can prove that it gives the same solutions as the Wright-Fisher process without mutation. Now, I think you might all heard about this name mutation. Mutation basically means how often the mutation rate means how often a genetic variant is changed to another genetic variant. Okay, and mutation can also have a back mutation to its original variants. The reason that I want to talk about mutation is that when we have intrinsic usage bias in what kind of variants we prefer to use, for example, even before I came to Australia, I might still prefer to use soda just because it has a single word, so it's easier for me to use. So I do have some intrinsic usage bias to use soda more often than the other two words. If we consider this into our model, then these people, the same paper that I showed you before, have shown that the solution of this model would converge to the same solution as the Wright-Fisher process with mutation. Okay, so the analogy here is mutation rate in genetics would be how often what is the bias for using one particular variance relative to the other. So now we have a picture of how one speaker in a community would update his memory and also update his language usage to produce more variance to the community. But what about the whole community? The whole community includes multiple speakers. So how can we account for that? Well, this has been first introduced by this Baxter and Bliss 2006 paper, and they called it a multi-speaker Wright-Fisher process. And so basically the idea is different people would have different degree of interaction, and the degree of interaction would determine how the usage frequency of one person would be influenced by the usage frequency of a variant by another person in the community. And different people might have different degree of interaction. So you might consider some speakers use language more similarly than the other speakers in the community. Well, obviously, this is the ideal model we want to use, but this model is highly complicated and it has no nice analytical solution. So it's not very helpful in terms of fitting to the real data. Instead, what can we do? We can introduce something called covariance structures into our Wright-Fisher process. And that is the key point in this new method I developed. Let me explain this idea using this diagram on the right. So in this diagram, the x-axis is the usage frequency of a variant. So it's bounded between 0 and 1. Okay, and the y-axis is the probability that is expected by the Wright-Fisher process. So we can see this curve, which means we would expect more chance that the person's usage frequency was false on the peak rather than false on the tails. Okay. Now let's consider a community where everybody interacts equally with each other. So let's call it a random interacted community. Okay. And we have a time axis. From time 0, we have some speaker born, and then they start updating their language usage just by chance due to the Wright-Fisher process. So each line shows how a person changes language usage or the usage frequency of a variant over time. And ideally, I should draw those lines as a zigzag because it's a stochastic process. But just for clarity, I just draw them as a line. Okay. And notice that during this time, some new speakers may be born. For example, this new speaker is born, and assuming that the new speakers randomly have random interactions with all the speakers in the community, then of course, the initial usage frequency of this speaker would also sample from the probability distribution of the Wright-Fisher process at this particular time when it was born. Okay. And then this newborn would again accumulate changes in its usage frequency over time follow the Wright-Fisher process. Eventually, at the present, the time we collect the data, we would also expect the whole community, their usage frequency would follow what would be expected by the Wright-Fisher process. So this is the ideal situation. But it's obviously not true. For example, a newborn may be more likely to interact with its parents. For example, if this newborn was the offspring of this individual, then the initial usage frequency of a variant of this newborn will certainly be same or more similar to the usage frequency of its parents. So its initial usage frequency would be here. And then assuming those 3 people are from the same family and they interact more with each other than the other people in the community, then this single family itself will start accumulate changes in usage frequency over time following a Wright-Fisher process. And eventually, at the present, instead of expect a unimodal distribution of the probability, we might see a multimodal distribution where, for example, this mode would correspond to this particular family. And how can we model this multimodal distribution? And a simple way is to using statistical modeling. So by introducing a covariance structures among speakers underneath our Wright-Fisher process. A covariance structure between speakers would, for example, if two speakers have very high covariance, it means those speakers would interact more with each other than other people in the community. And so they would have more similar usage frequency of a variant between each other than with other speakers in the communities. Okay, so that is the key point in this method. I hope I have explained this clearly. And if you have more questions on this core part, please feel free to ask me after the talk, or maybe just to shout it now. Okay, so now we have known how to account for non-random interactions in the communities. And how can we apply this to test the significance of the effect of one social factors on the changes in the usage frequencies in the whole communities? But the idea is very similar to linear regression. I think all of you know about the linear regression. So the basic formula is just, we have a response variable. It equals to an intercept plus a slope multiplied by a predictor and plus some error term, right? So in the traditional method, when we try to test whether one social factor has significant effect on the usage frequency of a variant, we often use a univariate regression, for example, a logistic regression. Okay. And in this regression, the response would be the observed usage frequency of one of the two variants of a speaker. So usually we only have two variants in those regressions, so that because the usage frequency of two variants have to sum to one, this reduces the problem into a univariate problem. So this is why quite often we only have two variants in a variable. And because it's usage frequency, so the null distribution would be logistic distribution. And this is why this is called a logistic regression. Okay, so the response would be the observed usage frequency of one of the two variants of each speaker. And the intercept would be the expected usage frequency of that variant. And for logistic regression, the expected usage frequency is simply the mean over all the observed usage frequency across speakers. And the slope would be the regression coefficient. And because we have two variants, so we need to interpret the slope in terms of changes in the usage frequency of one variant relative to the other variant. And then we have the predictor, which is a social factor. It can be a continuous variable, or it can be a categorical variable. When it is a categorical variable, for example, male versus female, we need to choose one category as the reference level. So for example, if we choose female as the reference, then the predictor has 1 if the speaker is a male and has 0 if the speaker is a female. And then the regression coefficient tells us the change in the usage frequency of a male by using one variant relative to the other and compared to the usage frequency of a female. So that's the key point, because we really need to make sure that we understand which level we have chosen as the reference so that we can interpret those parameters in a meaningful way. And the last term is often called a residual. Basically, we just assume it's the same variance or the same error across all the speakers. So all the speakers would have the same value for the error term. Okay, so hope most of you are familiar with the first part, the univariate regression that we often use. And the second part here in green is actually this new method, which is essentially a multivariate regression. And why it's called a multivariate? Because it allows a variable with not just two variants, but multiple variants. actually any number of variants. So the response variable has multiple columns, multiple variables, and each variable corresponding to the observed usage frequency of one of the multiple variants. And of course, we need to choose one variant as the reference variant, the same reason why we did this for the univariate regression. Okay, and the intercept again is the expected usage frequency of the speaker. And just like the univariate regression, now we want to incorporate our now model for language change, which is Wright-Fisher process. And if we also want to include intrinsic usage bias of a variant, we would like to use Wright-Fisher process with mutation. And then the coefficient and the social factors are the same, has the same definitions as the univariate regression. What is different and critical here is the difference in the error terms. So instead of assuming the same variance across different speakers, now we're assuming there is a covariance matrix among speakers. So two speakers with higher covariance will speak more similarly to each other than the other speakers. Okay, and not just having this covariance matrix, because we have these multivariate problems, multiple variants, so we also need to kind of embed this covariance matrix among speakers into another covariance matrix among variants of a variable, which is predicted from the Wright-Fisher process. So the symbol here, the circle with a cross, it's a mathematical symbol. It's called a connector product. Basically, it's just for each variance and covariance terms among variants, we multiply that terms by a covariance matrix among speakers. So we embed the covariance matrix among speakers into the covariance matrix among variants. And as a result, the distribution, the null distribution we assume for this multivariate regression is a Wright-Fisher process with mutation and with covariance structures among speakers. Okay, now it sounds quite complicated, but I'm going to give you some hands-on demonstration to show what I mean by those covariance matrices. Okay, so before I give you the demonstration, I want to first explain the differences between maximal likelihood and Bayesian inference because the method is in Bayesian framework. I hope some of you have already heard about the Bayesian inference. So basically, the common linear regression we use are all based on maximal likelihood approach, which means we want to maximize the likelihood of a model to increase its model fit to the data. So the definition of a likelihood is basically a conditional probability, the probability of our observed data given the parameters in our model. Using the linear regression as an example, the data would be the response variables and the predictor. And the parameter would be the slope, the intercept, and the error terms. Or we can use in mathematical terms as beta, y0, or sigma squared. Okay. And now if we plot x-axis as the data, here we have variable y and variable x, and y-axis as the likelihood, which is the probability of the data given our parameter. Then here, for example, we have three sets of parameters, the green parameters, orange parameters, and the blue parameters. And because the likelihood is the data given the parameter, so what we want to maximize is maximize the probability along this line, which corresponding to the actual data that we observed. And we want to find the parameter set that maximize this likelihood. And in these situations, of course, it is the orange parameter set. So the orange parameter set is called the maximal likelihood estimates of our model, which would gives you the estimates of slope, intercept, and error. Okay. So different from maximal likelihood, what Bayesian's framework cares about is called something called a posterior probability, or it's actually just a flip over of the likelihood. So it's the probability of our parameter values given the data. Okay, and using Bayesian's rule, we can derive that the posterior probability as just proportional to the likelihood multiplied by something called a prior probability, the probability of parameter without any data. So basically, we can incorporate any prior knowledge on the parameters into this probability term. For example, if we have a parameter, we know it has to be positive, then we are going to use a prior probability that is only meaningful for positive values. Okay, and I will show you some prior I will also show you some prior distribution I use in the model during the demonstrations. So now, if we assume the prior probability is just a uniform probability, so basically means all sets of parameters has the same probabilities before we collecting the data, then the likelihood is simply equals to the posterior probability here. So we have the same figure, but the y-axis now is the posterior probability. And again, the axis is the data, and the green line is the actual data we observed. Okay, but different from maximum likelihood, which want to optimize the likelihood along this gray line, we expand this gray line to a gray space, where the space is determined by the parameter values. So the probability of each parameter value given the data. And in this space, you would find that the orange curve, the orange parameter has higher posterior probability, the blue curve has the medium posterior probability, and the green term has the lowest posterior probability. If we continue sample parameter sets, then eventually we can fit a curve. And this curve is called posterior probability distribution. Okay, and if we integrate this code, so calculate the area underneath this code by integrating all possible parameter sets, this gives us the overall probability of the model. And this probability of the model is what I will use later on to explain what Bayesian's factor is. But don't worry about it too much now. So the next thing I want to talk about is how can we actually approximate this curve in a very efficient way. And this is by using MCMC or Monte Carlo Markov chain. And the reason that I want to talk about this is because there are some parameters that you might need to tune when you apply the R code. And I will explain it in the demonstration. So the algorithm I use in the code is called the Metropolis-Hastings algorithms. The basic idea is very simple. We start with the prior distribution of our parameter. So theta here is the parameter. And then we get some initial samples. Then we need something called a proposal distribution. So proposal distribution determined what is the next parameter sets that we want to propose. And an important parameter in the proposal distribution is called a tuning parameter. So tuning parameter basically determines how far this new proposal is from our old samples. Okay, we don't want this tuning parameter to be either too small or too large. Otherwise, the chain will take very long to actually approximate the final distribution. And this tuning parameter is something that you are allowed to change in the program. Okay, now, for example, we are at this proposal. And we also need to calculate something called acceptance ratio. Acceptance ratio is essentially the ratio in the posterior probability of, say, this new proposal compared to this old proposal. If this new proposal has accepted ratio, has a ratio in the posterior probability, this is large enough to accept this proposal, then we'll keep moving along the chain. If it's not large enough to accept, then we will simply discard this sample. Okay, so it's another important key things in MCMC, and I will later on talk more about this in the demonstration. If we continue sampling proposals over the chain for many, usually it's millions of iterations. Then eventually we would get a histogram of all these samples. And this histogram should approximate the actual posterior distribution of our model. Okay. So now I think it's the time to actually give you some demonstrations. And the code is in this link. And this is the paper that is currently under review, and hopefully you can get it very soon. And before I show you the demonstration, I want to explain the question that we want to answer. So first, of course, the practice data set here is the grungy creole data set. The question we want to answer is what social factor would affect the retention of the grungy variants or the heritage variants. And the factors we have generation, age, education level, exposure level to grungy variants, gender, also family and household. So family and household are now the factors that we are going to will influence the covariance matrix among the speakers. Okay. Now we also need to interpret regression coefficient. So the regression coefficient will tell us the amount of change in the relative frequency of using non-grungy variants relative to the grungy variants. So you can see here the grungy variants is now considered as the reference variants. And also we need to define a reference group because generation, education level, exposure levels, and the gender are categorical variables. So the reference group we have here is generation 1, female with high exposure to grungy and low education levels. So basically those are the female you would expect to use the most grungy variants. Okay. So I choose this reference group just in order to interpret the coefficient in a more sensible way. Now let's have a look at the actual code. So if you click on this link, you will find out this several README file. So it gives you this instruction on how to actually start using the code. The first step is to download all the files to a local folder. Okay, so I have already downloaded the files into my local folder. So those there are 4 files. Those are the files, and let's ignore those folders for now. Okay. Now the first step is there are a file called instruction. If you click on the file, you can see it gives you a step-by-step code of how to actually prepare the data and start the analysis and summarize the results. And let's first have, let's just try to run this a little bit. So the first thing we need to do, we need to library 3 packages. So the reason we need to library those packages because the code would use some function evolved into those R packages. So just copy and paste those lines. And some of you may not have those library installed in R. If you haven't do this, then you can type install.packages. And you can, for example, I want to install this package. Okay, and then you hit enter, and R will automatically install this package for you. Since I already installed all this package in my R, so I simply library them. Okay. And the next thing we want to do is we want to set our working directory to the directory where we save all these 4 files to this folder. Okay, so basically we set the path name. The path name, alternative way, you can simply drag these variables into your R console. Okay, so this gives you the directory. And then you set working directory and parentheses your password so that anytime you want to read some files or save some files, it's always saved in your working directory. Then we want to source the core functions for this method, which is in this function, Bayes bar rule. Okay, and I will go through this later on. And the first thing we need to do is we need to read in the raw data and prepare the data to a form that will be accepted by this function. So let's just copy and paste that. It's very quick. And before we do that, let's first have a look at the raw data. So this would be the raw data you want to prepare. These several columns, so each row is a speaker. These first several columns are social factors. So this columns is gender. So this speaker, because the reference group is a female, so this speaker is a female, so we have 0. These two speakers are male, so we have 1. Okay. The second columns is age. It's a continuous variable. The third columns is household or location. So for example, this person is 9, this person is 9, which means those two persons live in the same house. Also, similarly, we have family. We also have those. So this is generation 1. The first H is high exposure level to grungy. The second H is high education level in English. Similarly, this would be a person in generation 2 with high exposure to grungy and low education level in English. And the reason I want to combine generation, exposure level, and education levels in this way is because I want the model to be able to account for any interactions among those social factors. Okay. And for example, this person is actually a young people from third generation with a high exposure level and a high education level. So we have 1 for this person on these columns. And this person is third generation with low exposure level and high education level. So we put 1 here and put zeros in all the other group. Okay, so those are the social factor columns. Then we start with the language variable columns. So we have a V1, V1. So these two columns are for variable 1. And the variable 1 has 2 variants, 1 variant, second variant. And the second column, a row here, will give the type of the variants. For example, here we have 3 types, grungy, creole, innovative. And I mark those variants as 1 means it's a grungy variant, 2 means it's a creole variant, and there are, for example, here, 3 means it's an innovative variant. Okay, so variable 1 has 2 variants. 1 is grungy variant, 1 is creole variant. This person, it used the grungy variants, but not the creole variants in variable 1. So that's what this 1 and 0 means. Okay, so I hope it's clear for you on the raw data file. The other file is the variable file is only for plotting purpose. So it kind of a thing to remind yourself what is variable 1. So variable 1 is the meaning of up. And what type of this variable is, it's a production, lexicon, and a noun. Okay, so with these files, you can then plot those different variables with different colors or shape. And now we have read in this raw data, and we have transformed this data into a variable in R called the data. This data will then be used as the input data into the function being var loop. So let's have a look at what this data includes. So if you type ls, means list. So list whatever that is installed in this data. And you will find out, okay, data has 2 elements, one called lingua, one called seed, 0, type, and x. And lingua is another list. So here I will just show you the first 10 list. So what lingua is, for example, a cell 10, it contains all the columns in the raw data that belongs to variable 10. So basically, just all the values on those 3 columns and paste it into a cell, the 10 cells for the 10 variables. Okay, and so each row is a speaker, each column is a variant. What else in the data? We also have data theta 0. Again, it's a list. So I'll show you the first 10 cells. And this actually gives us the initial usage frequency in the community. So for example, again, In the tense variable, we have three variants. The first variant is a creole variant, so number 2 here, and the initial usage frequency is 0 because we choose a time point right before the English colonization. So in principle, no one in the community should use creole variants. So that's why the initial usage frequency of creole is set to 0. And for the tense variable, we also have 2 grungy variants. And because we don't have prior knowledge on which variant is used more often in that generation, so we set equal usage frequency as the initial usage frequency. Okay, so that's what data theta 0 is. So basically, if we go back, theta 0 basically is this one. Okay, and then what else in data? We have data type. Data type is also a list. So each cell corresponding to a variable, it basically gives you what type of variance each variance is. So for variable 10, we have a creole variance, grungy variance, and another grungy variance. Okay, and then data X is a data frame. That's basically the predictor matrix that we often use in linear regression. So each row is a social factor, each column is speaker. In total, we have 78 columns, so 78 speakers. Okay, so that's what the input data is. And now we need to determine enter. Enter, let's go back to the function itself. So enter means the number of MCMC iterations you want to run, or in other words, it's the number of proposal, how long the MCMC chain is. Okay, and here we made millions or even 10 millions samples. So here I set enter into a million. Interval is how many iterations, the interval that you want to write MCMC samples to a file. So for example, if I set interval into 100, it means at iteration 1, iteration 101, iteration 201, and etc., I am going to write the sample parameter sets into a output file so that later on I can read those output file back and start summarizing the results from the Bayesian analysis. And path name is basically the path of the folder that you want to save the results. So I use the same as our working directory here. There are a whole bunch of other parameters. You can set them as default, or you can actually change those values. So I'm going to explain the meaning of each of those values now. So to explain this, let me first explain what kind of parameters the analysis actually estimates. The first parameter is this M. It's basically the number of memory updates. So it's this parameter M. Okay, and because it's the number, so it has to be strictly positive. This is why I use a prior distribution as the gamma distribution, because gamma distribution is only meaningful for a positive value. And now we have some mean for the gamma distribution. Here, the default is set to 100 times, but of course, we don't know what the actual mean is. So we want to set the variance of the gamma distribution as large as possible so that we don't give too much information in the prior for this parameter. And the way to do this is by adjusting the rate parameter in the gamma distribution. And the smaller the rate parameter is, the wider the variance. So this is why you can see by default, I set a very small values for that. Of course, you can also change that. The other type of parameter is the intrinsic usage bias, which is this parameter here. So we have one value for each type of variance. And here we have grungy, creole, and innovative. And of course, you can also notate different variants, for example, in terms of morphological complexity, which is what we did in the language paper. And therefore, and in that way, your R variant type would be grungy, highly complex, grungy, middle complex, grungy, least complex, etc. And R would be a longer vector of parameter to estimate. And again, R has to be positive, so we apply a gamma distribution as the prior and a quite small rate parameter to increase its variance. And here, yes, it's also quite small in the default. The third type of parameter is theta. This is basically both theta. So it gives the estimated usage frequency of each speaker for each variant of each language variable. Okay, and I can show you the output. I have run the analysis before this, just to save time. So theta files would have 185 variables, so it will have 185 files. So theta 1 would be the estimated usage frequency for variable 1. It's a quite messy here, but once we read those files into R, it will be more straightforward. So basically, between two headers, this is one sample from one MCMC iterations. So variable 1 has two variants. So we have two rows, each row corresponding to one variant, and each column is corresponding to speakers. So we have 78 columns. So for example, this 0.49 is the estimated usage frequency of the first speakers in the data set using the first variance in variable 1. Okay, and of course, this is just a MCMC sample, so later on the estimation will be more accurate. Another important parameter, which is the covariance matrix among speakers we talk about, it's saved into this Omega file. And this file is quite big, so I'm not going to open it now. And basically, it saves a 78 by 78 matrix from each MCMC sample. And the last set is this beta regression coefficient. So it's a vector, so each element in beta corresponding to the regression coefficient for each social factors in our model. And we have talked about the interpretations of beta here, so I'm not going to repeat it. But one thing important is now the prior for beta is set to a normal because beta can be both positive or negative. And also we want to set the variance of the normal distribution very large. So here the default is 100. And of course you can make it even larger. Okay, so what else do we have? In the input, another input is called estimate.r. This is whether you want to using a right Fisher process with notation by incorporating intrinsic usage bias or without mutation. So this is just assume that intrinsically there's no bias towards any variance. By default, it's true. If you don't want to estimate those usage bias parameter, you can set estimate.r equals to false. And the remaining input I haven't explained are those tuning parameters. Those are the parameters we talked about here. So it determines how far your new proposal is to your old proposal. And you don't want this parameter to be too large or too small. So you need to do a bit of experiment to see which setting gives you a reasonable amount of increase or decrease in the samples in the posterior samples. And here I have some default. It works well with the grungy creole dataset, so maybe it will also work well with your own dataset. And another output is called this post. Basically, it gives all this estimation of beta, estimation of M and R into a file. And so each row would be a MCMC sample. This value, very big value here, is the log likelihood of your model. This is the prior probability of your model. And those are the samples of each of the parameters you want to estimate, including R and the beta. So I'm going to read those outputs so that you can have a clear view of what those are. So basically, once you have prepare the data, you can just copy and paste, but I'm not going to run it here because it's quite slow. It might take days, depends on the size of your data. So for 185 variables and 78 speakers, it takes about 1 week to finish the analysis. Okay, so now we want to read in our poster file. And then we can see the first row. So the first row gave us the log likelihood value, log prior values, regression coefficient of gender, of age, etc., the usage bias of G, etc. Okay. And the first thing you want to check, if you want to check if your MCMC chain has converged, the way to do this is simply plot the index of your sample with your posterior probability, which is simply the sum of your log prior and your log likelihood. So you will see something like this. And this part, the zigzag around a similar posterior probability, is what you want. So anything before this part, like this big drop here, is something you don't want, because this is when the MCMC is not converged yet. So it's not converged on the actual posterior distribution yet. So you want to throw this part away and only analyze the part that is sampled when MCMC is converged. For example, if we want to throw anything before 500 samples, we can do this. We can set a burn-in, and we can do that. And then in total now we have 643 samples from our posterior. Okay, so now we want to summarize the result. One result that we can get is using something called a marginal posterior distribution of the regression coefficient of a factor. So what marginal posterior distribution means? For example, here we want to only looking for the age effect. So the actual posterior distribution will be multidimensional, with each dimension corresponding to one parameter. And age effect, the regression coefficient of age, is one of those dimensions. But if we only want to look at the effect of age, we only want to focus on the dimension of age by integrating over all the probabilities in the other dimension. So this is called the marginal posterior dimension. In other words, it tells us the regression coefficient has over 99% chance to be negative. In other words, in all possible parameter combinations in this model, 99% of those parameter combination has age effect coefficient negative, which means no matter what happened to all the other social factors, age always have a negative effect on the relative frequency of using a non-grungy variance relative to grungy variance. Okay, so this is why we want to use a marginal posterior distribution to estimate the significance of a single social factor by integrating over all the other factors. So remember, we talk about regression coefficient as the amount of change in the relative frequency of using non-grungy variance to grungy variance. Therefore, positive coefficient means the social factor make the speakers to use more non-grungy variance relative to grungy, and the negative coefficient means they use more grungy variance relative to the non-grungy variance. Okay, so if we extract the effect of generation 2, for example, relative to generation 1, and then extract generation 3 to generation 1, generation 3 to generation 2, we can all see that it's almost certain that in every possible parameter combinations in our model, we're almost 100% certain that the generational effect has a positive effect on using non-grungy variance relative to grungy variance. In other words, younger generation always use non-grungy variance. And this is true no matter what the effects of other social factors are on this community. In other words, no matter how age affects the usage frequency, there is always a generational effect. This is consistent with our previous results, which show that generational effect, or maybe reflect a peer pressure effect, is independent to the age. It does not simply reflect a gradual change over time. It's a punctual change over generations. Okay, so that's the way to interpret those results. Similarly, we also found a positive effect of education levels in English. So speakers with more formal education in English using non-grungy variants more. And the certainty is almost over 70% across generations. Also, we found that the speakers live with grungy speakers use grungy variants more. And this certainty is lower than the education, suggesting that the exposure effect is relatively small than the effect because we have higher uncertainty in the effect of exposure. These two results are also consistent with our previous findings using a more traditional statistics analysis. But what is more interesting is after we have control for all these external social factors, what left us is the intrinsic usage bias. And we found that actually for our reference group, which is female generation 1 with low education and high exposure, we would expect those people to use more grungy variants, right? We did found that there is usage bias in favor of grungy variants than non-grungy variants in those reference group. This is why the relative usage frequency of creole relative to grungy or innovative relative to grungy is almost always smaller than 1. Okay, so how can we plot those marginal distribution? And these are the code to plot those. We're just going to quickly run this. Yeah, so that's basically how we get those plot. Okay. And I will using one example. For example, let's using generation 2 as an example. So basically, generation 2 would affect both HL2, HH2, and LL2 speaker groups. So the estimates of the coefficient for generation 2 would be the average of the coefficient for those 3 groups. That's why we have summed the coefficient of those 3 groups and divide by 3. And then we count how many times G2 is larger than 0. And this probability turns out to be 1 here. And then we can plot a histograms of those G2, and then eventually we will get this plot. So if we just plot this, yeah, so this is what we get. And the probability here is 1. So the probability that generation 2 has positive effect on using more than grungy variants is 1, so 100% certain. Okay, so that's marginal probability distribution. How's about the time? So we have 15 minutes left. I'll just quickly talk about one more thing. It's about the Bayesian factors. So Bayesian factors is basically the probability of the model with effects given the data divided by the probability of the model without effects given the data. And as I talked about before, this is basically by calculating the area underneath this curve. So we can use a Bayesian factor to very efficiently calculate the effect of different factors on different language variables. And the variables with higher Bayesian factors would be the variable that are mostly influenced by those social factors. So for example, here, determinators and the past tense turns out to be more variables more subjected to effects of social factors. Okay. And the code also includes the one to calculate Bayesian factor, and because I'm running out of time, so I'm not going to go to details of this. Okay. The last interesting thing is that we can actually plot the residual covariance matrix among speakers. So this tells us after controlling all these external social factors, are there any speakers that still speak more similarly to each other than the other speakers in the community? So ideally, if there are some strong clusters of speakers, we would expect some plots on the right. The plots on the right is actually generated from genetic data. So it shows you that people from the same geographic range would have more similar in their genomes than people from different range. So this is why those people in South Asia, they cluster together. In Asia, East Asia, they also cluster together. However, we didn't find any of those in the covariance matrix in our data, which suggested that there is no speaker clusters in the grungy creole communities after accounting for all the external social factors. Another interesting thing is that we can actually plot family and household. So each row and a column is a speaker, and we can plot their relative families and household. And we found out, for example, speakers from the same family or the same color bar here do not tend to cluster together. And this is another way to show that a family and a household has no strong effect on the clustering of language usage in the community. We can actually quantify this by calculating something called interclass correlation. And this is basically by extracting the correlations between speakers from the same family and plot their marginal posterior probability. Since I'm running out of time now, I'm not going to plot them here, but the result is in the paper, so you should feel free to check it if you want. The last cool thing we can do is that we can observe the relative fit of the model to our data by plotting the predicted usage frequency. So the data from those theta files to the actual observed usage frequency in the data. So the boundary data is not very informative, and therefore I use another data set, which is based on the counts. And you can see how similar, how matching the predicted and observed usage frequency are in those plots. I'll just quickly show you the continuous count data, how it looks like. So this is actually the data of Shall We data set that Felicity mentioned a little bit. So for example, in variable 1, you will see instead of having boundary 1, 0, we now have numerical values. And what this basically means is, for example, for this speaker, it has 16 chances to use the variable. 14 of them, they use the second variance, and 2 of them, they use the third variance. So this is the ideal data set that we want. It will give you lots of information so that your model fit would be very good, like shown in these figures. Just to mention that this pyramid means the variable has 4 variants. So each angle here corresponding to 1 variant. And the location of each dot is a speaker. They are colored by which village they are coming from. And the position of the dot in this pyramid shows its usage frequency. Okay. Yeah, I'm running out of time, so I'm not going to show you the code here, but it's all available. The last thing you might want to check is the code for discriminant correspondence analysis, which is available in this repository. Ideally, I want to also demonstrate this code, but because of time, I'll just show you where the code is. So it has 2 folders. One is for discriminant correspondence analysis. The other is for generalized linear mixed model. I'm not going to go through GLMM because that has now been incorporated into that new method, Bayes' bar rule. So we're only going to show this DCA folder. DCA folder has the code to generate 2 figures in the paper, and figure 2 is that colorful figure over here. And so you want to go to the folder of figure 2. And figure 2 has all this file. Again, to use this, you just download all those files into a folder. For example, I downloaded into here, DCA, and you click on the function. It gives you step-by-step instruction of how to make the plots. One thing I want to mention that DCA is an existing method. There is an R package to plot this. So you need to download this package from this link. And then you need to put the file, zip it, and put it in the same folder as this DCA folder, as your working directory. Okay, so it's in that folder. And then you need to source these things so that you can load all the function in this package. I don't know why this package is not published as a standalone R package, but this is, this is the way that you can use it. There's also several data that you want to prepare, and they have been, the format of the data have been explained very well in the paper, so you can read them. I'll just quickly show you the most important data. This simply gives you each column is a variant. So this is grungy variance of variable 1, creole variance of variable 1. Each row are speakers. 1 means they use the variant, 0 means they don't use the variant. The method also works for counts data. So if you have, say, 6 chances for this person to using variable 1, 2 is using grungy variance, 4 is using creole variance. So this is how you can plot those data. Okay. And yeah, so eventually you can get a plot like this. The plot has contained lots of information. So principal component 1 gives you the axis that explain the most variations in the usage frequency among speakers. And you can also plot those speakers onto the space and draw a 95% confidence interval eclipse that include the 95% of speakers in your speaker groups. So if those speaker groups, those eclipses overlap with each other, it means that those speaker groups actually have quite similar language usage with each other. So the factor here that different generations, so the red, yellow, and the blue are different generations, they are quite separable from each other along generation 1, suggesting that generation 1 is the axis that is mostly affected by generation effect. Okay. And similarly, principal component 2, you can see there is some divergence according to the education level. So education level would have a high contribution to principal 2. You can also plot the language variants onto the same space. And the way to interpret them is the variance that is closer to the eclipse of your speaker groups are the variants that these speaker groups use more often than the other groups. So from this plot, you can see a very obvious trend that the generation 1 tends to using grungy variants more than generation 2, than generation 3. And most of the creole and innovative variants are used exclusively by generation 3. Okay, so this is how we interpret this plot. You can, based on the contribution of different variants on the PC1, PC2, you can also calculate the overall contribution of a variable on each axis. So for example, here 92 has the greatest contribution. It means 92, variable 92 are used the most distinctively among different speaker groups. So 92 here is here. So it means generation 2 use, only generation 2 use the innovative terms for variable 92. And there are 2 creole 92 here. It means the generation 3 would use creole variants of variable 92 more often than the other generations. And there's also 1 grungy variants for 92 here. It means generation 1 only using the grungy version of the 92. So this is why 92 eventually is picked as the variable that is speaking most distinctively from the other among different speaker groups. So that's all I want to talk about. Sorry for the rush. At the rest of the time, what we left for questions. Oh, what variable 92 is? Yes, let me see. Variable 92 is additive. So it's a grammar.
Felicity, you're muted.
Sorry. For people who are trying to pin a bit of linguistic data onto this to sort of help make sense of it, the allative would be the difference between using the grungy variant, using the creole variant, which would be la or langa, or using an innovative form, which is this form ngiri, which is the result of phonological coalescence between a whole bunch of grungy allomorphs. So for any of the linguists, that's what it is. For any of you who are not linguists, that probably made little sense, but never mind.
Okay, so I'm sure everyone's heads are spinning now with all that fantastic methodology, and Jar's paper is on bioarchive, which is the preprint server that biologists tend to use. All that code is on GitHub. Hopefully, so this recording is going to be somewhere, I have no idea where, and maybe there will be also somewhere we can post links to those papers, links to their preprints, and links to the code. I don't know how we're going to do that, but let's work out how. And apart from anything else, you can email any one of us and we will answer whatever questions you can. Now, technically, we've only got 4 minutes left. I'm personally happy to stick around and keep chatting. So if anyone's got any questions, I'll just open it up first to see if there's any questions flowing on straight from Jar's presentation, and then we'll go back to that one we missed later. Has anyone got any questions they want to ask about anything in the presentation today?
I have a question for you, Jar, if nobody else is going to jump in. Was that anyone jumping in? Sorry, I didn't mean to. I promised everyone that you were going to say something about gender, but you didn't. But one of the reasons I was kind of hoping you would say something, and maybe you can, is because for the 3 generations, generation 1 being the eldest, generation 3 being the youngest, we don't have gender data for generation 2 because Cassandra and I were the people collecting the data, and it was inappropriate for us to be working with men of our own age. So I was wondering if you could say something about those gender results, given that there's a big gap in the data set.
Yeah, so this is why I only do the interactions among education's exposure and generation without including the gender. So gender is considered as an independent social factor to all these others. And so all the information from gender is from generation 1 and 3. And we found that female tend to, female is the reference, so male tend to using more non-grungy variants than females.
Yeah, and the reason that I say that is it's a very interesting result. It was something that came up in Catherine Travis's masterclass yesterday, and I'd forgotten that you had that result. So I'll pass that on to them because they're actually asking about gender. So there's essentially the male members of the data set are more inclined to use creole variants over grungy variants. When you say non-grungy variants, you're including innovative ones, I guess.
Yes, yes.
Yep.
Yeah. So that result is included in that paper. Maybe I can just quickly show you the plot.
Not that we're suggesting that the solution to language shift would be knocking off all of the male members of generations.
Yeah, so here is the gender effect. So the certainty is 70%. So it's kind of as strong as the educational effect. Over here, yeah.
Thanks, Shah.
I'll go back to the question that Bodine asked earlier, which is on the language endangerment work. So the question is, are the metrics for species endangerment roughly equivalent to the metrics for linguistic endangerment? How comparable are the two? So the way that endangerment is measured is completely different. So for species, you'll be looking at things like the critical factors in species endangerment, like whether you call something threatened or endangered or critically endangered, is to do with things like range reduction and population size reduction, and the correlates of extinction risk in species. They'll generally look at the aspects of the actual species themselves, like their reproductive rate or their body size or their habitat. And when we looked at languages, for a start, the scale of endangerment is very different because it's mostly based on transmission and usage. So domains of usage, like is it used in schooling, is it used in government? and transmission is, are the kids learning it, basically. And also the correlates that we will look at will be completely different. So whereas for species endangerment, you're looking at aspects of the actual species themselves, here we didn't consider any aspects of the actual languages. We're considering the kind of language ecology in which the languages exist. Do they coexist with a dominant language like Russian or English or Mandarin? Are they used in school? Are they officially recognised? Is there good documentation like other dictionaries and grammars, things like that? So the reason it's similar is because the way we ask the questions, the actual analysis uses the same machinery with tweaks. So JAR has developed these methods that are in some ways similar to the way we look at species endangerment, but they are also critically different, not just in the factors that cause endangerment, but also in the things we'll put in as important things in the model. So for example, for languages, we'll care about whether the distribution overlaps with other language distributions, whereas that's less of a factor for species. But the methodology, the things we have to care about, remember that example I gave you from New Guinea, where we have to take into account the spatial relationships between the areas we sample and the relationship between the languages themselves. So sorry, I don't know whether that answers your question. So the machinery is kind of the same in important ways, but the actual variables we use and the endangerment scores are very different. Also the endangerment for both species and for languages, not all languages, not all species have a kind of evaluated level of endangerment. So we can only use ones where somebody has actually evaluated that. And of course, the evaluations themselves might be out of date. So anyway, hopefully that paper is coming out this week. So feel free to get in touch if you need a copy of that. Has anyone else got any other?
Coralie has an interesting question, which we're actually working on. So Coralie, did you want to ask your question?
Yeah, sure. I was wondering if like a feature being above or below the speaker level of consciousness impacts that gender distinction you see.
Well, it's nice that you asked that question because we are actually working on this. So yeah, what you're referring to is, I guess, what sociolinguists call salience. And we talk about, I'm going to get, maybe you can jump in with the 3 different levels of salience, the Lebovian ones. Sorry, they've just dropped out of my mind.
Oh, it's been a while since I
Oh, no. Okay. And anyway, we're very interested in salience and sociolinguistic salience and what kind of, whether this is one of the drivers of language evolution. So we have experimental data that we collected this year from the grungy community, where people were looking at how salient they considered particular lexical items to be in relation to the different generations. So remember, Jar was saying that the different generational differences are one of the, sorry, I've forgotten, rather than age is a driver of language evolution here. So we're looking at salience with regards to generation, because this is the most significant of the variables. And we don't have anything to tell you just yet. We're just working through the experimental data with somebody else that we're working with, and we might have more to say about that next year. But it's an excellent question, and I really do think that sociolinguistic salience does have something to do with language evolution, yeah.
Cool. Now, I know we're running over time, but if anyone else wants to, and of course you can always just email us with questions afterwards if something occurs to you. Has anyone else got anything they want to, any points they want to raise? Felicity, do you want to read out that question from Sasha?
Sasha, yep. So Sasha says, well, do you want to, you can ask it, Sasha.
Yep. Well, I was just curious, for all of you working across disciplines, and there's obviously so many similarities and differences between linguistic versus biological evolution. So I'm wondering what you had to sort of become aware of in the process about what assumptions you were making and what the differences were and things like that.
For me, that's actually been one of the great things about doing interdisciplinary stuff, because for biologists, we're always working with the same evolving system, right? DNA-based organisms. So we don't know how much of what we assume in evolution has to be that way, whether it's a fundamental property of evolution, and how much is just happens to be the way that, you know, organisms evolve. So to give you an example, if you read any biology textbook, they'll say evolution depends on random mutation, meaning mutations that just happen without respect to whether they're needed or whether they're useful. And that's absolutely true for organisms, for species, for genomes. But it's not true in linguistics. In linguistics, some change, some variation appears randomly, and some can be deliberately brought in. You can invent a new word. You can deliberately adopt a new word into your vocabulary if for social reasons or for explanatory reasons or whatever. So then that's a nice contrast. You can say, well, if we relax that assumption about random mutation, does that change the way things evolve? Can we use the same models? What difference does it make? So for a biologist, this is one of the reasons why working with language evolution is good, because as you said, it absolutely fundamentally challenges you to think about the nuts and bolts of evolutionary change. I would say that the key to cross-disciplinary work is patience, for starters. It takes a long time. I'm very, very lucky to have worked with people who can patiently explain things to me, because as you know, when you hear it for the first time, it kind of washes over you. And can I just say, I have lost count of how many times Felicity has had to explain what an ergative is to me, and I still don't understand it. So there's some things I will never fully get, but the patience to explain things. And I have lost count of how many times Jar has had to explain multidimensional statistics to me, and I'm still not really there. So having a team where on the one hand you can all explain things to each other, but on the other hand you can trust in each other's expertise. So I can say, right, I don't get what an ergative is, but I know that you do, so I'm going to park that one with you. Sorry, I don't know whether that was but I should say that interdisciplinary stuff, you have to accept it takes longer, because there's so much to learn and there's so much that you have to work out between you. So it's not like you can just pick stuff up and run with it in the same way you might be able to do with something within your own discipline. So it does take a time investment. Jar or Felicity, do you want to add anything to that?
Yeah, just same thing, that this started with a lot of conversations. And Sasha, you're one of the summer research students who are part of this work. Those conversations started before that summer research project. And then I went away and in talking to Lindell and Jar, trying to figure out what the data set needed to look like, because we had the corpus, but not the data sets, right, for the analysis. So there was a big conversation that went on before that. Simon Greenhill, I believe, was our cherub, and Lindell still has some of the paper bags that she was writing on to understand all of this. And then, for instance, I went back, and then, we had you mob as a wonderful crew of summer research students who then created the data set. And that was still only 2017 or something. It was still a long time ago. So it's taken a long time, because this stuff is so new, to get from, that initial conversations, the data sets, to the sort of methods that Jar's talking about. So, yeah.
And another thing to say is that it's actually can be very hard to publish, because by definition, you're doing something new. And so cross-disciplinary stuff is hard, because if it goes to one set of reviewers, they're not going to get the other side. And so it really is, you have to sort of be prepared to invest for the long haul. But certainly I've found this some of the most intellectually exciting work I've ever done, because you're constantly having to learn new stuff. So I don't know whether any of you, having listened to all this, your brain is kind of hurting, having heard all of these different things. Well, that's how I feel like most of the time. But I think that's a good hurt,
Yeah, awesome.
Yeah, absolutely. Yeah.
Cool.
All right. Is there anything else? I'm mindful of not taking everybody's Saturday up too long, and I don't know whether you've got other masterclasses to go with to this afternoon. Is there anything else, or shall we? I just want to say again how much I wish we were all standing in a room together, because that's the way to really get these conversations flowing, and that's the way that those interdisciplinary links really build up. So if you're from an institution that has both evolutionary biologists and linguists, see if you can actually get some of those conversations started. A lot of the time you'll have a nice cup of coffee and then it won't really go anywhere, but other times you'll talk and the germ of an idea will start, and it will take a long time to follow that through. But if you start those conversations, if you're open to new ideas and new ways of looking at things, then yeah, more brain exercise and hopefully some exciting new frontiers to cross.
Yeah, and look, I think it's these kinds of masterclasses. Good on you for attending. It was, we originally sort of came up with this because I had attended a class that Lindell was giving, I think in 2017, and was listening to it and had an aha moment that was finally something that I thought that we should be dealing with language data sets. And then, as I said, Simon Greenhill played cherub and introduced me to Lindell and Jar. So I think these sorts of things are really important to get those brain juices going and just to think about questions and how you can answer those questions by using new methods.
So the summary is: be bold, be brave, be inventive. Don't be afraid to go where no linguist has gone before or evolutionary biologist. Keep talking to people, talk to us if you've got ideas, talk to each other and get your brain spinning. And when you see a new way of doing stuff, have the courage to see if you can follow that path along. Cool. All right. At that stage, I'll say thank you very much for coming along and sharing your Saturday with us. I hope that in future years we'll manage to all meet in the same place and continue this conversation. And thanks to Felicity and Jar for being such excellent people to work with on this. And I hope that for somebody out there, this is also the start of new ideas and new projects as well. So thanks very much, everyone, for coming, and hopefully we'll catch up in the real world sometime. See you later.
Thank you. See ya.
Did the three of us want to stay on this, or after everyone's left?
We can have a brief conversation. I'll then have to run off.
I'll stop recording.
Yes.
No, yeah, do that.